Good morning, good afternoon. Welcome to the 2022 International Kleefstra Syndrome Family and Scientific Conference. For those of you all that don't know me, my name is Jeff Ryan and I lead our effort at IDefine. I want to lead the conference off with expressing gratitude. Gratitude for those who helped plan today's event, for those speaking today, for the attendees, and perhaps most importantly at all, the entire Kleefstra Syndrome patient community. I am incredibly excited about today's discussions. It's about where we've been, where we are, and the potential of where we're going. A couple of quick housekeeping notes. Please drop all of your Q&A questions into the chat or into the Q&A box below. And as time allows, we will follow up with each speaker on the questions. And then there's already been a couple of questions about whether this conference will be recorded or not. And you should all have received notification that it will be recorded. And then we're going to edit the clips afterwards into individualized speaking sessions and present them to the community as soon as they're ready. So as I mentioned, today's going to be, it's going to be a lot of information, right? We heard, we had a great conference last year. We took some of the feedback, we distilled it down, and we're having a little bit of a shorter session this year. So each speaker's been asked to speak for about 15 to 20 minutes. And not get too technical, but obviously when we're talking about some of the scientific presentations, there will be technical conversation that if you do have questions, just please enter them into the Q&A. We're going to talk about the work that's being done right now. We're going to talk about our data collection efforts. We're going to talk about some of the clinical efforts that are going on and the readouts. And then finally, we're going to talk about IDefine and what our initiatives and objectives are for 2022 and moving forward. With that being said, let's begin today's conference. Leading off today will be Joost Kummeling. He's going to be giving a speak about the natural history study that's ongoing. And so, Joost, I will turn it over to you. Thank you very much.
Thank you. Thank you very much, Jeff. I hope I will share my right screen at this moment. Let's see. I think it's this one. If everything is right, you can now see my presentation. Yes, okay, that's good. So, thank you very much, Jeff, for introducing me. And thank you all for joining in this very exciting conference. It's exciting for me as well. Today, my name is Joost Kummeling. I'm a medical doctor and a PhD student working under Professor Kleefstra in the Radboud UMC in Nijmegen in the Netherlands. And today I have been given the opportunity to speak about my research, and today we'll give an update on the natural history study that I'm currently conducting. And let's see. So Tjitske Kleefstra is sort of the main person regarding the Kleefstra syndrome research here in the Netherlands, and she has gathered a team together in the Radboud UMC, the University Medical Center. And that team consists of multiple people with all their own expertise, and their expertises range from Kleefstra syndrome research in mice, Kleefstra syndrome research in brain cells in fruit flies, and I am one of those clinicians who focus on Kleefstra syndrome in humans. My work is more or less a continuation of the work that has been done by Carlijn Vermeulen. She's a children's and youth psychiatrist who works here also in the Radboud UMC. And she wrote a thesis about psychiatry in Kleefstra syndrome, which I'm going to tell something more about. And what she saw is that people with Kleefstra syndrome may experience several psychiatric disorders like major depressive disorder, psychosis, regression, which is a loss of functioning, and which can be, in fact, is a loss of certain skills. It can be motor skills, can be communication skills, and also people with Kleefstra syndrome may experience problems with sleep. And you can imagine that sleep problems are maybe the most debilitating, the most horrible problems regarding the whole family, because if a child in a family doesn't sleep, the rest of the family doesn't sleep, and if I miss one night of sleep, the next day I will not be functioning at 100%. But let's say if I do not sleep or sleep very badly during a whole week, well, I'm almost a different person after 1 week. So it's a very important problem that that may occur, and we want to tackle that problem as well. Often the major depressive disorder or psychosis can present in a different way in people with Kleefstra syndrome than in other people than other clinicians are used to, like which Carlijn Vermeulen already said with major depressive disorder, that often a major depressive disorder does not present as a typically sad mood, but more as an irritable mood. So these kind of things, as they present differently, it makes it quite hard to diagnose and therefore also hard to treat. And we do not have standard treatments ready for these kind of problems that may occur. This is the main thing that I want to tackle, and I'm going to explain to you in this study. So in 2019, we set up a patient study, which has a follow-up of 4 years. We want to follow the participants for a couple of years to see how they're doing. And we are collaborating with multiple centers around the world to make this all happen. And we started in April 2019. So it's now almost April 2022. So we've been busy for a couple of years. We have some data, but we're not there yet. The first objective we set out to fulfill is to understand the natural history study of Kleefstra syndrome, and we want to know its natural course. We want to know every aspect of it. What is it? What does it mean for the patient? What does it mean for the family? But secondly, we also want to understand the regression that is observed in Kleefstra syndrome and the effectiveness of olanzapine, because this is a sidetrack. Olanzapine is a drug that is mainly used in psychiatry, mainly on people with psychosis, like schizophrenia. And Carlijn Vermeulen saw that the olanzapine can have quite a positive effect on Kleefstra syndrome patients who experience regression or psychotic disorders. Although this is promising, it is not yet confirmed as such. For the inclusion of the study, we included people with a Kleefstra syndrome diagnosis, of course, from which we were able to obtain a written informed consent and who are 12 years of age or older. This is because the main psychiatric problems that we want to look at occur later in life, so not really in childhood, but after 12 years of age or older mainly. There are some exceptions, but I will not go into that. But we want to focus on 12-year-olds or older. And to map all the different aspects of the Kleefstra syndrome that we want to learn from the participants, we set out to establish a test battery specifically designed for this patient group. And this test battery consists of questionnaires and neuropsychological and psychiatric tests. And for you, it's important to see that in the bottom line, those are the names of the tests that we are conducting. And we have, as part of the questionnaires, we ask the parents / caregivers of the participants things like we want to know about the participants' communication skills, we want to know about their daily functioning skills, and we want to know about their social skills. And to give a few examples of the questions that are in the interview, does the communication-wise, does the person know the meaning of at least 10 words? Can the person indicate a preference if allowed to choose between things? Like daily living skills, is he or she able to eat with a spoon? Is he or she able to make his or her own bed? And regarding social skills, we ask questions like, does the person express two or more recognizable emotions, such as happiness, sadness, fear, or displeasure? And can he or she keep a secret for at least 1 day? And these three things that I just mentioned, the communication skills, the daily living skills, and the social skills, collectively, we call that adaptive functioning. And the adaptive functioning is the main outcome of the study. It's the main thing that we are looking at. In addition to this, to adaptive functioning, we look at sleeping problems that may have occurred or may occur, any mood disorders like depression, or any symptoms of psychosis that may occur. And with the neuropsychological tests, we look at signs of autism. We look into the learning and memory capacities of people with Kleefstra syndrome. And in addition to that, we also look into the visual motor integration, which is sort of a fancy word for the collaboration between the seeing of things and in a reaction to that movement of fingers of hands. And this is a sort of an overview of all the tests we do. So this is quite a complete test battery where we've worked to establish. And how does this work in practice? Well, we follow, as I told before, we follow the patients for a couple of years. So when we've included them in the study, we do the tests, so the questionnaires with the caregivers, and the tests with the participant. And after a year, we do the same thing, and after another year, we do the same thing, and year after year, we do the same battery of tests. And we want to see if there are any differences over time. So, are there any? Does any regression occur, or are there any other problems that occur, like mood disorders or such? And in the case that there might be a psychotic disorder that arises, we advise people to start olanzapine. That's the antipsychotic drug that I told you about a few slides ago. We cannot do this by ourselves because we all are in the Netherlands and it's a pretty small country. We can do quite a bit, but we need collaboration around the world, and therefore we also started a collaboration with Boston Children's Hospital, who have started to gather patients as well, and they've included quite a substantial amount of participants for the study. And in the beginning, we calculated that we needed around 60 people with Kleefstra syndrome of 12 years of age or older to participate in the study, to have a decent bulk of information. And our idea is to have to include 40 patients here in the Netherlands and 20 patients somewhere else, so in the rest around the world. And in this case, Boston Children's Hospital is sort of being on the forefront there. And this is the update on the study. So at the moment, we are at 39 inclusions in the Netherlands, and Boston has made 13 inclusions on their side for the rest of the world. So in total, we are at 52 participants with Kleefstra syndrome in our study. Of those 52, we've split them into two groups, and it's maybe a bit more technical, but we split them into the early detection cohort and the late detection cohort. And what this means is that in the early detection cohorts are the people who have been without any problems, without any psychotic disorders, or who have had psychotic disorders but were included within 2 years since the start of the symptoms. And sorry, I have to take one step, one moment. And in the late detection cohort, we have Kleefstra syndrome patients who have had psychotic disorders already, and were included more than 2 years before, more than 2 years after the first symptoms arose. And this is the main state of affairs at the moment. So in the early detection cohort, we have 32 people now, and in the late detection cohort, we have 20 people. And at this moment, we have to say that inclusion has stopped. We're sort of at full capacity, and because of the whole Corona pandemic, which has influenced all of our lives, we were not able to get the 60 that we wanted to have beforehand. And We expected to have 15 people in the early detection cohort to be using olanzapine because the start of psychotic problems. But at this point in time, there are 9 people in the early detection cohort using olanzapine. Because we did not get to that 60, we are looking into sort of alternative analysis to get the most out of our data, and currently that seems quite promising. And in addition to this, of course, we have started up a psychopharmaco survey, which can be found on the kleefstrasyndrome.org website, but also on the IDefine website. And this is in addition to the study to get as much information as we want, sort of to help us with our first objective, sort of to understand the whole natural history of the Kleefstra syndrome. This survey is intended for parents or caregivers. It's of course about the Kleefstra syndrome patients who have or an EHMT1 variant or a 9q34.3 deletion. And in contrast to the other study that I mentioned, this survey has no age restriction. So we want to gather as much information on as much people with Kleefstra syndrome as possible. And with this survey, we are hoping to collect several things. It consists of 7 parts. We wanted to gather general information on the participant, but also medications that are used, which ones are effective, which ones aren't. We want to know if there are any regression periods, how sleep is going, and if there are any comorbidities, so if there are other health problems at the moment. If it's possible, we also want to collect information about the treating physicians, so we can contact them and ask them for more information about the person. And of course, there's also some room for remarks if there are any remarks regarding the survey. A bit of a practical aspect now. The survey itself, it takes about 20-30 minutes to complete if you want to take a look. And a tip I can give you is it's important to gather relevant medical information before starting, because otherwise we will be asking you, of course, a lot of things about medication, all kinds of medical records. So it's important to already have that handy. Up until this point, we actually have 33 surveys completed, so that's quite a significant amount. But we were hoping to gather more with your help. And when you start the survey, make sure you finish, because sometimes people sort of forget they started up and they think, okay, it's a lot of work. It's a bit more work than I thought, and they close it. So we have sort of empty files, which is not really handy for analysis. But yeah, when you start, please make sure to finish. And this, what I said before, this survey is available via the kleefstrasyndrome.org website or the IDefine website. And I will give a link to the survey at the end of the presentation. So I'm reaching the end of the presentation already, and there are some key points I want to give you. So good to know that here in the Radboud UMC, we are currently working with a strong team with expertise on multiple disciplines. And we want to tackle the diagnosis and treatment issues of the psychiatric disorders in Kleefstra syndrome. And despite of the COVID issues we have had, the study is doing quite well. We managed to still keep on gathering information during the whole pandemic. The inclusion has been closed at this moment, and I'm hoping to get results to you soon. And especially to the families, I wanted to let you know that you have to be aware of the sleep disturbances and regression, as sleep disturbances may be a sort of precursor for a loss of skills. And you can contribute to the study via the survey that I mentioned. I want to especially thank all the people from the Radboud UMC who are part of the whole clinical team on this study. I want to thank all the lovely people from Boston Children's Hospital and all the people from IDefine who've made this conference possible. And have given me the opportunity to speak here. I'm obliged to thank also these people from the Noun Project. I used some of their images during this presentation, and of course, lastly, I want to thank all of you for listening to me and hopefully paying at least some attention. So thank you very much. Here's the QR code for the survey. If you want to participate, you can sort of aim your phone at it and go directly to the survey, or maybe save it for later. And well, if you have any questions, please, I think it's wise to ask them in the chat, and we'll try to answer a couple of them. Thank you very much. And I will now stop sharing.
Thanks, Joost. That was, as always, super informative. And I wanted to try and cover a couple of the questions that are in the chat. And again, if everyone can just continue to put any questions in the Q&A and utilize the chat to kind of have conversation between each other, that would be super helpful. One of the questions, and I'll open it up to use, but if any other panelists would like to weigh in, certainly please feel free to do so. But the kind of question was, have you seen KS patients that are exhibiting psychosis having conversations with themselves? I've seen anecdotally evidence of this, but have you all seen clinically any experience with this?
Well, as I mentioned earlier, it's quite difficult sort of to have a good idea of a psychosis in a person who is experiencing Kleefstra syndrome. If a person without the developmental delay, without intellectual disability has a psychosis, it's sort of you can easily detect it, since that person might communicate to others like I feel like I'm being followed, or indeed talking to other people in the room who aren't there. And because there's a wide variance of development within the whole Kleefstra syndrome group, there may be some people who have the right amount of communication skills to actually have sort of conversations with people who aren't there. And I've seen a couple of patients who have actually showed psychotic symptoms like that. But on the other hand, there are also people who have with a really different subset of symptoms. Yeah, but it's indeed it can occur. Yes.
And then there's a question, and I want to provide a little bit of context just from my vantage point, but there's a question around olanzapine, Zyprexa, and CBD. And I would just kind of take a step back that, ultimately, these are all kind of treating symptomatic challenges of the condition, right? Like there is a big push on our behalf, and I know as well as the team at Radboud and further about let's get more granular and kind of find root cause, right? And so we can treat symptoms as much as we want, but there is probably a shelf life where you start becoming, something will either wean off, you become accustomed, your body and biologically shifts and starts not responding the same way. And so, we want to continue to drill deeper. And that's where a lot of the focus on our research efforts comes in. We can use some of these off-counter or readily available things, but they're not necessarily customized to Kleefstra, right? So I think that's an important footnote to have here. And then as it relates to CBD, I did put something into the chat. If there's interest from the community regarding CBD and just getting more information, we actually have a member of our SAB, Dr. Allison Mootri, who has presented on the effectiveness of CBD in some neurodevelopmental conditions. And so it is worth discussing. Just please indicate your interest to us and we can take it from there. One other question real quick, Joost, is will folks be automatically included into the second study or sign in there as well? It says they're in the first study that you mentioned. So do they have to reach back out, or are they automatically transition into the secondary study?
So the secondary study is mainly for the people who aren't who didn't have the opportunity to join us in the first one. So we have at least our means are sort of limited, and we could only include so many people with the means that we have. But we still received a lot of questions around the world from people who wanted to participate in one way or another. And for those people, we've set up this specific survey so that they can contribute in their way to our research, and we can get information from them as well, which we will benefit. We hope we will benefit them from them, and we hope that you can benefit from them as well.
Great.
Yeah.
All right, Yus, thank you very much. We will now transition into the next discussion. And again, for everybody that's putting questions into the chat, we will record all of those if we're not able to get to them live today. We want to be kind of respectful of time as we move through the program. So please continue to put questions in the Q&A and not into the chat box. It'll be a little bit easier to monitor from our behalf. I'll speak for myself there. So next up, we will move into Dr. Chitska Kleefstra. She's going to be providing a discussion around the topic of differing perspectives of EHMT1 and Kleefstra syndrome, and also alluding to the effort around the guideline development that is underway and hopefully going to be concluding within the next year. So Dr. Kleefstra, take it away. Oops. Okay, so my error. These are the joys of live conferences sometimes. She has not signed on just yet. So why don't we move into, we'll have Dr. Neil Nadif Kasri join in. So Dr. Kasri is going to be presenting a discussion around the topic of leveraging stem cell technology to dissect the cell type specific contribution of EHMT1 and Kleefstra syndrome. So Dr. Kasri, I'll pass it to you.
Thank you, Jeff. I hope you can see my slides. Can you see my slides? Yeah, presentation?
We can.
Okay, perfect. Yeah, thank you very much. Well, first of all, for this putting this fantastic conference together. I think it's always an honor to be able to share our research, our plans with you, our strategies on how to work on Kleefstra syndrome. So my name is Neil Nadif Kasri. I'm a professor at the Radboud University, and I work together with Chitske Kleefstra, Annette Schenk, Hans van Bokhoven, and also Joost on Kleefstra syndrome for many years already. And particularly my lab is trying to understand really what is happening now in those nerve cells of patients with Kleefstra syndrome. And that's actually a very difficult task, because essentially the human brain is inaccessible. So if we really want to understand what is different in a patient's brain, then one would like to be able to enter that brain to look at what is different. We want to manipulate it. We want to test some hypothesis, and so forth and so on. And it's obvious that this is not possible with nobody's brain. And so one has to find other ways to do that. And so for the many years, researchers have found surrogates for the human brain, basically by investigating animals' brains which have actually the same mutation as which we can find in patients. And this has been very helpful and is still very helpful. But obviously, a mouse brain, for example, is not the same as a human brain. And so what we set out for a few years ago already is to really try to understand how the nerve cells of Kleefstra patients function, and how they potentially function differently than healthy controls. And so for that we make use of stem cells. And today I just want to share with you our strategy, how we use them. And probably many of you have already heard about stem cells. and how we can use them, but I really want to take you step by step along the way. So what we can do is actually, we can take for any individual, we can take either blood or fibroblasts or hair cells, and we now have a possibility to convert those cells back to stem cells. And basically stem cells are, you can see them, the cells that are at the origin of life. And so they have the capability of becoming any cell type that you want. And so having them in a dish in the lab allows us now to choose to which cell type to differentiate them. And obviously, as I mentioned, we are interested in brain cells. So actually in all the cells that are present in the brain, those include nerve cells, but also non-neuronal cells. And so here you see an example of those beautiful nerve cells. I always show this because it always gets me excited. You see the complexity of those nerve cells. And this is an example of after 1 month in such a dish, you can start from the stem cells and you can get these very complex cells. And so in green, you see in principle all these dendrites, we call them. And on those dendrites, those cells receive information from other cells. And the spot where, the place where this information is arriving is actually at all those red spots that you can see along the green dendrites. So there's actually an invisible other nerve cells here that makes contact with this green cell here. And so this is how information is basically transmitted in the brain, but then not with few cells, but with billions of cells. And that's now we have the possibility to have this in addition, to study this in addition. So if we zoom out a little bit and we look at the bigger picture, what we actually want to do is being able to start from patients, generate these stem cells, which you call induced pluripotent stem cells. And then from those stem cells, we generate all types of brain cells. And I think today we'll hear more about it from other speakers, but I will really be focusing on brain cells themselves, on nerve cells. And once we have them in a dish, obviously, we want to do something with it. We want to measure something. We want to be able to measure something so that we can say, oh, this is different than the healthy control. And I will show you the way we do this. And that's what we actually called phenotyping. And once we have a phenotype, we try to use this to understand how this phenotype comes about, but also we can use it, of course, to try to test all type of compounds that are available to see whether we can bring it back to the normal situation. And this, obviously, the last part is the most interesting, but also the hardest part. So how do we now measure? What do we measure from those nerve cells? So as I mentioned, we can generate those nerve cells now in dish. We can compare nerve cells between Kleefstra patients and controls. And something that is really important to mention is that now every nerve cell that we generate starting from a particular patient has exactly the same genetic information as the donor. So it means if a patient has a certain mutation in the EHMP1 gene, then that exact same mutation will be present in this nerve cell, in addition to all the other differences in the genetic background. And so this is what we do, but what can we then measure? Now, what we know is that those nerve cells need to communicate with each other. So they need to talk to each other. And so we know from many disorders, including Kleefstra syndrome, that often that's actually the problem. It's the communication between the nerve cells that seems to be different. So we like to grow those nerve cells in what I call a smart dish. And that's a dish that has electrodes embedded in the dish. So this is not a real image, let's say. How it really looks like is what you can see here on the left. So you can see here and zoom in actually of the electrodes, which are indicated in this as this black dots. And the structure that you see here, this gray structure that you see here on the background, are actually the neurons that are the nerve cells that are growing on top of the electrodes. And so if we put it very schematically, we have this grid here of electrodes, and then we can grow the cells on top of the electrodes. Now, the nice thing is that, as I mentioned, the neurons, they like to communicate with each other. And so we can start to pick up this communication through those electrodes. And so that's exactly what we do. And so here on the bottom, you see a color map and the changes in color. And the more red you see, the stronger the communication is happening. And the bluish, then you have no communication. So we can measure directly actually how nerve cells from a particular patient or person, actually how active it is and how it communicates with each other. Now, obviously, we are scientists, and we want to be able to monitor that a little bit more quantitatively. And so what I show you is a typical example of how those measurements look like. So we call this a raster plot. And so every line that you see here is basically one of those electrodes. And if you go from left to right, you have time. And so every vertical line that you see now, those black stripes, is every time that a neuron actually communicates with another neuron, another nerve cells. And so once in a while, you see that they actually all communicate at exactly the same time. So this means that what we call the cells are in a network, and cells like to be in a network, just like people like to be in a network. And so a healthy population of nerve cells will actually always show this typical pattern. And analyzing this in detail really allows us to really ask the question, when are those nerve cells communicating? How often they are doing this? With who are they talking to? And also how loud are they talking? And so this gives us really a lot of very valuable information. And I really want to re-emphasizing, this is really, we can do this now for every individual patient. Now, the nice thing is then or the most, I think, interesting aspect is that we can now compare a control population with the Kleefstra syndrome patients. And here you have an example of 3 healthy controls on the left and 3 Kleefstra patients on the right. And without having to understand anything of the complexity of this kind of experiments, I think if you look at the left side, you see very clear similarity between all those controls. You see that there is the graphs, let's say, look very similar. However, if you look to the right, you see that the graphs look quite different. And mainly, you see differences in the boxes that I've highlighted. You see that this is a very long black box. So there are moments of very high activity, and then there are moments of very low activity. And that's very different than what we see in controls. Now, we can do this with very simple cultures with very simple contribution of nerve cells, but we can also make the whole preparation much more complex. And one very important idea in neurobiology is that within our brain there needs to be a balance between excitation and inhibition. And without going to detail, I think you can probably imagine that this is important and that you don't have too much excitation but not too much inhibition, and this needs to be in balance. And so we can also measure this using exactly the same type of approaches. And then we make the whole neurons a bit more complex. We don't generate just one type of neurons, but we generate multiple types of neurons. And then we can do exactly the same exercise. And then again, we can look at this very complicated plots. And what we see now is that by doing those type of experiments, we can actually really pinpoint which cell types in the brain are actually contributing to this different type of communication. And we have started until now with very simple, with very few cell types, but we are increasing the complexity. And of course, as we are increasing the complexity, the whole research becomes more complex, but we also are getting then more to the real situation of a human brain. Now, to summarize a lot of work, I use this slide. In basically on the left, one can say that in healthy controls, there is a certain balance between excitation and inhibition. And what we found in Kleefstra patient cells, so in the dish, I have to really emphasize this, that we see that there is more, it leans more to excitation. There's more excitation. And we think that it is because the inhibition is somehow not working very well. Now, what can we do with this? As I mentioned, we are measuring this really in a dish, and we just started a really exciting project together also with Chitske, where we are now trying to correlate what we find in a dish to what we actually can also measure in the real patients, because this is really important. And how can we compare this? Well, you might all have heard of EEG measurements. Those are actually ways to measure also activity of the brain. You can put this kind of cap on, and then you can look at signals that are very similar of the signals that we can measure also in a dish. And so this project will really aim at measuring both the activity in the dish and the activity of that same patient, so the real activity. And so we try to compare those. And the whole project is we really call it the multi-level strategy, and I think it's really, really important because we don't want to measure only something in a dish and actually not knowing what we are measuring. So we, again, from the same Kleefstra patient, we are getting blood samples of fibroblasts. We are generating those cultures, and then we are measuring this excitation inhibition balance at different levels. And so what is really important is to be able to compare that with real measurements of the patients, of their brain. And why are we doing this? Well, once we have measured this excitation inhibition balance, so the phenotype, we can now try to, of course, rescue this. And what's interesting, there are actually quite some medications on the market that are not being used for Kleefstra syndrome that can impinge on this excitation inhibition balance. And so with this strategy, we aim at least to get to uncovering at least certain potential routes for therapy. What does it mean also? I think that this means that I want to end with this so that we can use now stem cell based experiments to assess individual brain cells from individuals. And this is really essential because we know that there is quite some individual variation at the clinical level. And so we should not assume that even though patients have the same type of mutation, that the functioning of their misfunctioning of their nerve cells is similar across all the individuals. And so stem cells really allow a strategy to now test that at the individual level. And obviously this type of approach can also be used for trying to predict the potential of new medication, which of course would then need subsequent validation in more complex model systems. With this, I would like to end and thank, of course, all the people in my group that have worked on this and contributed, to people in Nijmegen, and of course the whole team of IDefine that has made this Congress possible. Thank you very much.
Thank you, Dr. Kasri. And then there's a couple of questions in the Q&A that we wanted to cover, but I wanted to also kind of extrapolate on a comment I made earlier about a lot of these drugs are just being for treating the symptoms around KS, and that's not to minimize the opportunities that exist that you kind of just referenced, that there are opportunities for drug repurposing currently out there. And Dr. Kasri, I know that we have discussed before about how in some of your studies, the electrophysiology readouts have had a condition like kabuki kind of overlay with what's going on with Kleefstra in kind of neuronal activity, which is really interesting. And so perhaps that's a good segue into a question here about what are the medications on the market now that might help treat the excitation?
Yeah, we have tested in this system. So we have tested certain antagonists for very specific channels that are FDA approved, and I think it's still early days, because what we, as I mentioned, so we can test this in this kind of culture system, but what you always want to do is really go in a more complex system, and so we are doing this at this moment. So but there are actually quite some compounds or medications available that work on all this excitation inhibition balance. But one really needs to find, there is not just a compound that either increases or decreases excitation. So it's much more specific than that. So one needs to really pinpoint it very well and be very careful for that. So and that requires a lot of research and time. And so we now have some idea of what kind of molecular target we should go after. And that's what we are investigating. So I can't tell you readily okay, this drug will do it. I wish I could, but I think the strategy to get there is laid out, and I think that's important.
Yeah, I think the Brain Model Consortium, what's being conducted there is going to be incredibly important. It's not going to be a study just to kind of level set expectations with the community. It's not going to be a study that happens overnight. It's going to take some time, but there is some really important work being done there.
Yeah, I think I say that we have scheduled 6 years to do this. So just to give you, and that's a reality. We have to get the cohorts, we have to make the stem cells, we have to record the EEGs from those patients, we have to do all the work in dish, and then try repurpose drugs to see whether we can rescue certain, and then if that's possible, we might go to certain end of one studies. But yeah, this will take time, absolutely.
Yeah, and I think there was some commentary in the chat around collecting biosamples, and just to kind of reiterate or add in extra context that those biosamples are what is utilized to help create these iPSC lines. And so I don't know that we could have too many. We want to see, with KS having such a variance, it's important to be able to view the full spectrum, so that when we get to the point of screening a therapeutic, that we can see how it interacts across the spectrum. One other quick question for you, Dr. Kasri, from my buddy Haluk. He asked, will these drugs need to be delivered directly to the brain? And if that's the case, how will they be delivered?
Yeah, this is really a very good question. So, because when we add something into dish, that then we don't have the blood-brain barrier, so we don't have this barrier that prevents drugs to get in. So that's very easy. Then we always have to look at the specific drug, whether it's known that it crosses this barrier. If it crosses this barrier, then we are in luck, let's say. Then there's no, it will reach the brain. If not, there are either chemical changes that need to be made, or there are also ways to get it directly into the brain, which is of course more difficult and more cumbersome. But basically, it will have to be evaluated at the individual level, and I think, yeah.
Great. And then I just saw a question pop in about what year is the study in, and if that's in reference to the brain model study.
Yeah,
this just launched. So the team just received funding, and it's around 8 specific genes, but Kleefstra is certainly one of those, EHMT1.
Yeah, and there is a website, brainmodel.com, we will we will update there or the progress of so feel free to follow it there, but also of course via IDefine will keep you up to date.
Great. Thank you, Dr. Kassery.
You're welcome.
All right, so now we will move into the discussion from Dr. Kleefstra. So Dr. Kleefstra, are you there?
Yes, I am. So
wonderful.
Trying to share the screen now. Let me see. Do you see the screen?
We do.
It's a full presentation, or?
Right now we have you and I spotlighted, but we'll spotlight you alongside the presentation.
Okay. Okay, so thank you very much, Jeff and team from IDefine and the Boston team for organizing this this webinar. And I'm sorry I was a little late because of again this time challenges and change, but we are there, and I'm glad you already heard the great stories from Joost and Neil. And I thought to add on that and give you a flavor of several other studies or projects that are ongoing in our team. And I think with the 10 years experience that we have now in following up cases and including novel cases that are seen in our hospital, we gradually get some experience to have a more systematic approach of several angles. And so we are based in Nijmegen, as you've already heard, in the Radboud UMC, that's a university hospital. And there, for Kleefstra syndrome and many other syndromes, we have a great team, clinical team, and this is embedded in what we call the Expert Center for Rare Congenital Neurodevelopmental Disorders. And basically, there is 4 large angles that we approach for Kleefstra syndrome and other syndromes, and that's from the medical side, so the clinical part and clinical genetics. It's important. The biology, so we try to closely work and connect to underlying biology, as Neil just showed very nicely by studying these cells from the patients. And we closely collaborate with psychologists and psychiatrists to understand the developmental issues and neuropsychiatric aspects that a lot is common in these syndromes, and of course also the Kleefstra syndrome. And by integrating the knowledge from these fields, we try to optimize the personalized strategies to get better management and intervention for your children and the patients we see. And we are happy to have this great team and wonderful, bright young researchers, clinical researchers among our team, and I show you here, and I will pass them during the presentation. And I think Joost here on the left, you have already heard, and I will not further comment on his studies, but I will show some other studies, and it might be some difficulty there or technical difficulties, but I try to pass you through, and there might also be time afterwards to discuss. So focusing purely on the molecular aspects in genetics and related to the clinical part, I'll first show you a glimpse of the studies that Dimitris is conducting. So you may probably have heard about it, that the most of the variants we identify in ETMT1 in the gene by our diagnostic approaches, they are one type of variants and called loss of function. That's a typical, the vast majority of variants in ETMT1 causing Kleefstra syndrome belong to that category. And until Dimitri started these studies, there were only a few missense reported. So that's another category, and we call them missense changes published in literature or known to us, and that were in fact only 3. But by the current day's technology, so meaning the whole exome sequencing or whole genome sequencing strategies, there's many such missense variants identified, and we receive a lot of questions from worldwide from colleagues or patients or parents that send us a message about a variant with uncertain significance, in short, VAS, and that mainly are mostly confined to these missense variants. So we started to collect this and approach that by a systematic manner and try to learn from the spectrum of VASs that is now out there and known to us. You can see schematically how the gene, E71 gene, is composed of exons. So the exons are all these vertical bars, and this comprises the entire E71 gene, and it's located in the tip of the chromosome 9q arm. So this is the entire chromosome 9, and this is the q tip. This is E71, and this is in fact also you see here all the exons with the vertical bars, but here's also protein domains schematically presented, and on the top you see the different variants that were known to us in 2012, so 10 years ago. So we collected missenses, a lot of such VASs. You again see the schematic presentation of the protein of E71 with the protein domains colored here, and above you see the different missenses that we collected. So we collected molecular information, but on top we also collected clinical features, of course, and we used 3D protein modeling. So how, because E71 protein is not a vertical bar or a horizontal bar, but in real life it's really a 3D, makes a 3D structure, and variants, E71 variants or mutations can change such a structure. And we used an additional test. I'm not sure some of you have heard of it, but it's called the DNA methylation signature. So we can look at different signatures on top of the DNA sequence. We can also look at how DNA is packed with proteins with a specific category, and that's called the DNA methylation. And nowadays it's known that for E71, for Kleefstra syndrome, there's a specific DNA methylation signature, and we can ask the laboratory to look at it, and that can confirm the diagnosis. And this study we did in collaboration with Bekin Sadikowicz from Canada. So for this 29 missense, we used these 3 approaches, and in the end we could provide for 26 of them concordant results, meaning that we could classify 9 as benign variants, are not causative for the problems, no Kleefstra syndrome, and 17 of them were definitely pathogenic, causing the phenotype and underscored the Kleefstra syndrome presence in the cases. But 3 variants were discordant, so were not in line with Kleefstra syndrome and not with the DNA methylation signature, but neither were classified or could be classified as benign. So for these 3 variants, we needed additional confirmation, and for that we started collaboration with a Japanese group. I'm also happy they are here today. They will present not on this work, but they also carry out this real great functional assays, and they have taken that up in their essays and more look by a biochemically manner and see whether these variants could provide pathogenic effect through different means, actions, than the classical E71 missense or loss of functions. So that's an ongoing story, but to summarize this part, so the variant interpretation currently, variants that derive from whole exome or whole genome sequencing can be challenging, and it's not always easy to classify them, but our data, I think we are now quite comfortable that we can combine the clinical input with the 3D protein modeling studies and the DNA methylation studies, and probably functional assays for those that cannot be classified by the combination of the first 3 means. And we also have indications that there is different functions for each MT1 on top of the histone methylation that is usually thought to have a major function. So that's on the clinical, genetic, and molecular aspects. Then another study, and that's in fact coordinated by Ariane Bouwman, another bright PhD, started in our group last year, and she will also be a PhD working on this brain model that was just presented. And she's more looking at clinical profiles, so looking at the EEG measures, but also measures that can be done in the body composition, in blood, in urine, looking at metabolism, etc. And for those who were present in the webinar 2 years ago, our pediatrician Joyce Geelen presented the study on growth, and this is just a summary of the findings by that time, where we have seen that the growth is typical for this syndrome. So this is a plot, the age on the x-axis and the y-axis is the standard deviation. So it's a statistical way of showing data. And here in gray, you see the mean from our Dutch population. And here, cases start to grow, but you see there is, if you look at the height, that's this curve, height increases, the standard deviation it makes a short top already pre-puberty, and then compared to the mean Dutch population, there was a decrease in height. If you compare, it's relative, but the weight behaves differently, in fact oppositely. So there it gradually increases with age compared to the mean in the Dutch population. So of course, for the individual cases, it can be different, but as a group, as an entire group, the Kleefstra syndrome population has a relatively relatively shorter in height and larger in weight compared to the mean, and thus has also increased BMI at puberty and adult age. And because of this growth patterns, we also started to more systematically measure other functions as thyroid functions, and also the X-ray to define and evaluate bone age that you can read from X-rays. And what we see quite frequently, I must say, is a defect in thyroid function. So this is a very schematic representation of the thyroid functions in our body. So the thyroid produces this thyroid hormone, and it's under influence of the thyroid stimulation hormone from our brains. And when the thyroid hormone it decreases, the thyroid stimulation hormone increases, and the other way around to get this feedback loop correctly. So when we find a low thyroid hormone, then usually then you would expect a high TSH, but we see in several cases that it's not that clear. So we see more a normal TSH or something on the high normal range, but not that increased as you expect. So, but we do see quite several of the patients a low thyroid function, and we need to supply the patients with the thyroid medication. And regarding the bone age, so in those cases there we started to have bone age measured, and there is this automatically measurement that our radiology department has, and I think many university hospital will have. It's called Bone Expert. But we not only saw a bone age that might be some ahead of the biological age, but we also saw in quite some cases a low bone mineralization, and also some mild bone abnormalities, and that warrants us now to provide and to make in every child such hand photography. We in the past we did not do that in a systematic way, but we do now, because it might be relevant, give relevance for the clinical guidance and clinical follow-up. So to summarize this more or less clinical part, so we call it the clinical characterization and do it in a better and more specified and systematic way. And Ariana Baumann, so the young doctor, PhD studying this, she is very happy if you would connect to her in case there's any abnormality found, or if you have questions on these measures or would like to have such measures done, and we can see if we can be of any help there. Then the biology is just shown, not very nicely presented by Neil, but it's great work he's doing on the cellular models. In Nijmegen, there's also this great work done by Annette Schenk. She's studying fruit flies as a model organism, also for Kleefstra syndrome, many syndromes. And one of the projects that we are doing together with Annette is on sleep. And that project is done by Lara van Rensen. And sleep is very difficult to study. There's many aspects that play a role in sleep. But it did also pass during Joost's talk, where sleep might be a precursor for severe neuropsychiatric problems. But of course, young children might also have sleep difficulties, and it's often very complicated. But one of the questions is whether the biology behind sleep plays a role. And that might be true for each MT1. So for the Kleefstra syndrome, yeah, the biology, so the processes in the brain might be underlying at least part of sleep problems. And we know that might be present in other syndromes as well. So that is, Lara is focusing on sleep, and she studies that heavily in the Drosophila, because fruit flies also sleep. You may not be aware, but they do sleep. They have a rhythm. And in fact, this is the setup that can be studied in using fruit flies. So here you see the 24 circadian reflection of the circadian rhythm, 24 hours, and the bar diagrams that are indicated, measured by sleep per 30 minutes. And when gray is normal, you can see that in the yellow line reflects severe problems in sleep and showing less sleep for that specific fruit fly model that has been studied. And there is many parameters that is comparable that can be studied in Drosophila and that we study in human as well. So comparing sleep parameters and sleep trajectories for fruit flies might give us information on the biology for our human situation, which is obviously much more complex than such a small model organism. So Lara is conducting sleep studies in different syndromes, and among them the Kleefstra syndrome, and we ask the Dutch parents who participate to have this calendar filled for 2 weeks, and also a sleep questionnaire to be filled in. And for those that appear clinically appear abnormal on the inventorization of these data, that we can follow up if needed in the sleep clinic that we collaborate with in this study. And of course, it's not always sure whether your child is asleep or awake. If you sleep yourself and the child doesn't make that noise, you're not completely sure. And therefore, we extend the studies now with measures like the motion wards and an MFIT, which can be put under the mattress during sleep. And it can also measure all kinds of parameters. So also here, we for some sleep measures, we could easily include people remotely by sending out questionnaires and sleep calendar. And we have given consent for that by our ethical committee. So if you would like to, well, to learn more about it or to have participation, you can email send an email to this message. Well, then Neil already nicely presented the work. So that's the ongoing study that we just started, this great effort to really trying to combine this multi-level approach for the individual patients, measuring the EEG, but on top of that, we need a detailed medical history, physical examinations, neuropsychiatric assessments, and the measures on the metabolism, because all these measures influence, of course, the condition of the individual. So that we try this in this huge effort to combine. Then all studies and all the experience and all the data that we collect and get knowledge about, I think we are there now to head to get it more formalized in a guideline. And we are happy that we are supported by the European Union to develop such a guideline. I think there's around 55 professionals who are really active participating. And I'm also thankful to IDefine, who organizes work group meetings for such a guideline. And there the aim is to get this international standardization and recommendations for diagnostics, the care and the management for Kleefstra syndrome. And also here, I really need to thank Ariana Baumann, who's really taking the lead, the clinical lead to get the work done. And I also put the rest of the juniors over here, or who all have a very important role in getting the data and getting the work done for this guideline. It's really a huge effort that we need to do together. And I also like to thank Joyce Galen, the pediatrician, who is tightly involved in the care of these children in our hospital. And with that, I would like to give the floor back to you, Jeff. Let me see if it works. Yes, there I am. Not sure if Jeff.
I'm here. I'm having a little bit of technical difficulty. And there we go. So thank you for that. And now we can jump into the chat for a couple of questions real quick. And they're pretty wide ranging right now. So let me start with one question here, Dr. Kleefstra. It said, could medications, particularly olanzapine, be responsible for the weight gain in some of the older KS patients? Dr. Kleefstra, I think you're on mute. Sorry.
Yes, you're right. Yeah, so in general, it is known that olanzapine or antipsychotic drugs make you vulnerable to increased weight. But in our experience, because we also give clear instructions on the food intake, we not see so much weight gain on olanzapine. So, and also the curves I showed were corrected for olanzapine or antipsychotic weight increase. It's just intrinsic. It's really intrinsic in the metabolism of the syndrome.
Great. Yeah, I think diet is a whole nother kind of can of worms that we could get into. Diet and GI and
diet.
And again,
I had only 20 minutes. I think I already passed it. I think so. We also studying gastrointestinal problems, the diet, indeed, how it's called, microbiome, and there's so much to study. And cardiology is also very important. So, yeah. So this I would just give you a flavor of all the work that's done. It's really a lot of work done, and I think that's the most important message.
Yeah, I think just to that point, there is so much going on throughout the kind of Kleefstra syndrome effort right now. It's going to be hard to really tap into all of that today, and hopefully, these conversations, like the one that we held last year and the one that we held this year, is kind of giving you a high level overview. But there is a cadence of communication that we want to develop via our social media channels and via our newsletters, where we really ping the community about getting a little bit more granular in more webinars and whatever those touch points can be. We are open to providing that communication and want to provide that communication. So whatever the kind of relevant topics are for the community, we want to be able to touch base on. So one other question real quick, I'm going to try to combine a couple of these questions. One is regarding, is there a correlation between bones and teeth in terms of thyroid function? I've actually seen quite a few posts recently about teeth and the enamel being soft, but also whether or not calcium supplements could potentially be utilized to help offset osteoporosis.
Yes, they might be favorable. It's difficult to have a general recommendation at this point, or to dive into individual cases, but I think my message just be aware. We recognize there might be bone problems, and not necessarily clinically already obviously. So there need not to be fractures, for example, because you might expect there might be bone fractures or increased bone fractures, but I don't think that's our impression. But yeah, there might be real to severe mineralization problems, and that's just check with your doctor and make an extra X-ray if not yet done.
Yeah, and then so I think we can wrap up this discussion for now. I think maybe it's more of not a wrap up, more of a pause. And then again, a lot of these questions, I think what we continue to find is that we are a rare condition where there's still so much to be known. And, that's going to segue later into some of the data collection efforts. I think that from our community, or from our perspective within the organization, we talk about data a lot, and it's because we want to really collect all of this information to be able to start really formulating a stronger idea of what's taking place, and not having these kind of one-offs, but understand what are the overlaps, what are the themes, what are the things that we're consistently seeing. And without patient population contributing to that through these data collection efforts, then we're never going to get to the point where we need to get to, right? So it's really an intertwined kind of relationship where it's so much, it's very reciprocal. So thank you, Dr. Kleefstra. Please just keep your eye on the chat for me, because I'm sure there's going to be quite a few questions coming in for you. And next we are going to shift to, there's been a couple of conversation, or a couple of questions already put in the chat regarding what we kind of deem as therapeutic window. And so the next conversation will be speaking to that. And just a little background here. Interestingly enough, these folks first came on our radar last year, the Riken Center out of Japan. And their most recent paper that we were really kind of blown away by as a community was in reference to the therapeutic window and the potential reversibility of Kleefstra syndrome. And in a serendipitous turn of events, the article was published on Kleefstra Syndrome Awareness Day. So sometimes the stars align in the universe. And so we thought it was really a profound paper, and I'm looking forward to the presentation from Dr. Shinkai and Dr. Yamada. So we will let them take it from here.
Hi, I'm Ayumi Yamada from Riken in Japan. I'm so happy to join this meeting today. The title of today's talk is Reversibility of Neurological Phenotypes in Kleefstra Syndrome Model Mice by Postnatal GLP Supply. I will talk about our research published in iScience last June and want to introduce my unpublished current work. Probably I do not need any introduction for Kleefstra syndrome. So our group identified the G9A and GLP complex, which encoded by the EHMT2 and the EHMT1 genes. And this complex mediates the methylation on histone H3 lysine 9 residue, and this methylation regulates the transcription. After Dr. Kleefstra group reported the EHMT1 gene is responsible for Kleefstra syndrome, we decided to study this disease too. So now we are analyzing the pathological mechanism and the possibility for fundamental treatment of this disease. So other group reported the EHMT1 heterozygous knockout mice and the brain-specific knockout mice showed the behavioral phenotypes. So we are also using our own EHMT1 knockout mice as a model mice for Kleefstra syndrome. First, we examined whether the expression of GLP protein can rescue the behavioral phenotypes in EHMT1 knockout mice. To think about the future clinical trial, we induced the GLP expression after birth, and this experiment allowed us to examine whether epigenetic information can be altered in terminally differentiated neural cells by postnatal GLP supply. And furthermore, we want to elucidate the molecular mechanism of this disease. So the EHMT1 gene is essential gene for all of the cells in our body, but why deletion of EHMT1 gene causes the neurological phenotype so strongly? Today, I want to talk about this 1 and 3 mainly because many of the patients' families are joining this meeting today. So these are the genomic structure for EHMT1 knockout mice and inducible EHMT1 transgenic to express GLP protein. For knockout mice, we deleted the set domain, which is responsible for the methyltransferase activity. And for EHMT1 transgenic mice, we cloned the EHMT1 cDNA followed by the CAG promoter in Lothar 26 locus and put the stop cassette in between the LOXP site at upstream of EHMT1 cDNA. By combination with the Cure recombinase transgenic mice, we can control the GLP expression by locus and the timing. And we use these 3 Cure lines, and 2 of them are Cure ER lines. This line can induce the GLP by drug injection. So we can analyze when and where we need to express GLP to treat EHMT1 mice phenotypes. So this is a movie of one of the behavioral tests called the open field test. We put mice in the box and how see they behave in the new environment. This test can examine voluntary activity and anxiety. So wild-type mice is curious to the new place, so it immediately starts moving around. In contrast, the EHMT1 knockout mice just keep sitting at the corner and move like this. So as you see in the previous slide, the total distance traveled in 20 minutes test was shorter in EHMT1 knockout mice compared with the wild type. And this really number shows how much the mice are curious to the new environment. Here, the number of knockout mice is less than wild type. When GLP was expressed in whole brain from embryonic stage, both results are rescued by GLP expression shown in green. The rearing number was small in knockout mice, so anxiety could be high in EHMT1 knockout mice. So we examined anxiety using light and dark test. So the EHMT1 mice showed anxiety, so they are not going into the light area, so the time in light is smaller than wild type. And this phenotype is also recovered by GLP expression. So from this result, if GLP was expressed in whole brain from embryonic stage, we can rescue the behavioral phenotype in EHMT1 knockout mice. And this result also suggested the reduction of GLP in brain induced this phenotype. Next, we examine whether GLP-D expression after birth can rescue the phenotype. We use caretakers system to express GLP in all cells in whole body. So GLP was induced by tamoxifen drug administration at juvenile 3 to 4 weeks old. After that, we waited 4 weeks and analyzed the behavior at 8 weeks old. And after that, waited another 2 months, and we did the second round test at 16 weeks old. First, I checked the GLP expression in the brain by Western blot. You can see the increased amount of GLP protein in transgenic mice line. And we examined histone H3K9 methylation level by immunohistochemistry using the neuron marker NeuN and the astrocyte marker GFAP. Both in neuron and the astrocyte, K9 methylation level was increased by GLP expression. Using these mice, we performed the open field test and the light and dark test. At 8 weeks old, we could not see any recovery by GLP expression in open field tests. But another 2 months later, in 16 weeks old, both total distance and the rearing number were increased, and wild type in blue and knockout mice with GLP expression in green showed no significant difference statistically. In light and dark test, the time in light was also elevated at 17 weeks old, but we cannot see such effect at 9 weeks old. So this is a summary for this experiment. So the expression in whole brain from embryonic stage, we can recover the behaviors. And the expression in whole body from juvenile stage, it took time, but it can improve the behavioral phenotypes. So this is the experiment using knockout mice, but it suggested the possibility for cure of Kleefstra syndrome. Next, we want to know which cells are required to rescue phenotype. We tried the expression of GLP in neural cell of frontal brain. Like GLP induction in whole body, we induce GLP expression at 4 weeks old and examine the behaviors at 8 and 16 weeks old. But as you see in graph, we could not see any improvement even at 16 weeks old. So here's a summary. The GLP-D expression in frontal neuron are not enough to rescue the behavioral phenotypes in EHMT1 knockout mice. So GLP supply in non-neural cells would be required. But we wondered whether no effect at all in GLP supply in neural cells. So we analyzed the histological phenotypes in EHMT1 knockout mice. Well, not only the behavioral phenotypes, EHMT1 mice showed the histological phenotypes in brain. It's been reported the spine numbers in EHMT1 knockout mice is less than wild type. And our mice also showed the decreased spine number. In addition, we measured the spine head size. The the head size in knockout mice is smaller than wild type. This indicates the knockout mice spines are immature. Why spines are less in knockout mice? We analyzed the microglia in the cortical region and detected the number of microglia is significantly increased in EHMT1 knockout mice. And these increased microglia were active home. So it suggests that active microglia prune the spines from the dendrite in knockout mice. We saw the activated microglia in EHMT1 knockout mice, so we speculate neuroinflammation was induced in knockout mice brain. So we examined the expression level of inflammatory-related genes. We detected the RNA level of caspase-1 and the downstream factor IL-1 beta were elevated in knockout mice cortical area. And from single nuclear RNA-seq analysis revealed that caspase-1 is increased in excitatory neuron and the microglia in knockout mice. We examined these histological phenotypes were recovered by neuron-specific GLPV expression, and we can see spine number and the microglia number and caspase-1 expression were recovered by GLP expression in neuron, but spine size is still immature. So we are thinking this could be the reason why neuron-specific GLP expression cannot improve the behaviors. If case phenotypes were caused by neuroinflammation, these phenotypes could be reversed by inhibition of inflammation. So we prepared the EHMT1 and IL-1 beta double knockout mice and performed behavioral tests. Contrary to assumption, you see the result in purple. Knocking out of IL-1 beta cannot rescue KS behaviors. We cannot rescue the behaviors, but same as neuron-specific GLP expression, IL-1 beta can recover the microglia number and the spine number, but still spines are small and immature. So this result indicated duration of IL-1 beta pathway can reverse the histological phenotypes, but it's not enough to rescue the behaviors. This is our current model of EHMT1 knockout mice brain. Because of the decreased amount of GLP, the histone H3K9 methylation was reduced, and the inflammation-related genes are upregulated. This induced the microglia activation and followed by the excess spine pruning. These inflammation and other factors lead to the defect of neural network, and KS mice showed behavioral phenotypes at the end. The data I showed today is the result using model mice, so we want to know whether neuroinflammation occurs in human patients. And we know that inhibition of inflammation is not enough to rescue. We need to elucidate other pathways involving KS phenotypes. And we know we need to stimulate the reconstitution of neural network to rescue the phenotypes. So now we are trying the environmental enrichment. This environmental enrichment is reported to induce the reconstruction of neural network and the neurogenesis. So now I will show the very preliminary data that I'm working on right now. So we choose the voluntary wheel running as an environmental enrichment. From 3 weeks old, we allow mice to play with running wheel for habituation, and then from 4 weeks old, we put wheel in each cage and allow mice to run freely. After 4 weeks, at 8 weeks old, we analyze the behavioral test. This is still preliminary data, but as you see this graph, the mice with wheel group, there is no difference from total distance between wild type and the EHMT1 knockout mice, and the anxiety phenotype in EHMT1 mice was also recovered by wheel running. Now we are analyzing what was changed by wheel running and how long it can keep this effect. So today's my talk's take-home messages is our data suggests that case phenotypes in mice can improve even after birth. So there is a possibility to treat brain dysfunction in KS if same finding also exists in KS patients. So these are the collaborators to help this project. So that's all. Thank you so much. Hi.
Thank you, Dr. Yamada. That was fantastic.
So
that was really, really great. I'm not even going to try and answer one of the, or try to recapitulate one of the questions in the Q&A. I'll let you maybe answer that once we go offline. But I think, I'm seeing the information come through the chat right now that it's just been an absolute blessing to be connected with your team over the past year, and we can't thank you enough. I think what the take-home message provides us is something can be very fleeting at times, and that's hope. And that's, we want to see what we can do to propel these efforts, to give you and the other researchers the tools that you need to further the effort. So I don't see many more chats or questions in the Q&A, but please stay around, and if you can answer some of those questions in the Q&A, it would be very appreciative. But on behalf of our entire community, thank you so much for the work that you're doing.
Thank you.
Take care. Okay, so now we are going to, again, that relationship, when they were kind of discovered by us, they've been working on Kleefstra syndrome for roughly 15 years, and we just didn't know about them. And that was really interesting to find out. And so as soon as we did realize that this group was out there working on this, we reached out and connected, and have since been just developing that relationship. But Dr. Kleefstra and the team and Nijmegen has been in contact with them for some years now. So there is a huge collaborative effort going on, and it's amazing. It's amazing to be able to see what the potential is for our kids. And as I say, hope can be fleeting at times. It can be dangerous for us, because we all want what's best for our kids. But certainly when you see studies like that, it's hard not to get excited about it. So we will now transition to someone else who also has been working on Kleefstra syndrome for some years now, but that from IDefine's perspective, Eric Sheff and I just recently connected with, and are also very excited about the work he's doing. And so Dr. Carl Ernst is going to be joining us next on a discussion with the topic of modeling EHMT1 deficiency in human brain cells to identify new pathways for potential treatments. So Dr. Ernst, good to see you again. Take it away.
Hi, everybody. Thank you. It's nice to be here. Can you see my screen?
We can.
Okay, let me start my. So I guess first, I would have seen or spoken to some of you families out there in Boston in 2016, and just that was a really important meeting for me. It was very nice to meet you. I'm sorry we can't be in person now. And it was really nice to meet your kids then and connect with the other researchers. So thanks, Jeff, for getting this going. So I want to share, we've been working on this for about 5 years or so now, and using, you know, cells from the 9Q community. So thank you. We cannot do this without you. You're looking at a picture here of, you know, brain cells we made from P, you know, active cells. You can see them firing in addition. Just gives you a sense of the kind of things we can do with these cells. All right, so this is going to be a bit, I kind of want to get to my point, so I will sort of throw it at you. I will keep it as little technical as possible, but I have to go there a bit because I think we have some pretty interesting discoveries that I want to share with you. So just first off the bat, this is our population group. We have 2 patients, one of which is a full gene deletion. That's the one on the left here. And then on the right, we have a missense mutation. And this one was actually in Dr. Kleefstra's presentation. I saw it. It's in the anchorin repeat domain. I'm assuming it's the same subject. And each one of them is matched to their sibling. And we do that just to make sure we can really reduce the variation in our experiments. So our basic approach is to make, you know, just like you heard from Neil, and I will not repeat all that work, but, you know, we have a similar design. We make stem cells from blood, skin, or pee. We turn those into stem cells. That's sort of what you're seeing here. And then we can turn the stem cells into a progenitor-like stage. And then these progenitor cells can be turned into neurons, just as you saw from the Netherlands group, and you can do different things with these cell types. So I was very interested in, you know, Jeff has really hinted at this today. I'm very interested in the absolute first direct effects in EHMT1 to see if we can mine down to the root cause of the disease. At some level, everything else we see is going to be a secondary, tertiary, or an indirect effect of the initial problem. So it's not to say it's not related, but it may not be the core root. So this has obsessed me for the past few years. And we did a lot of work in stem cells themselves and the neural progenitor phase, and then the neurons as well. I'm not going to share all that data with you, but we did the obvious experiments of looking at, histone modifications and, gene expression patterns. So what I want to share with you is the robust, I think, discovery we made from one of these we basically did, throw the kitchen sink at the problem, see what comes up, look for the most interesting, potentially important finding, and chase it hard. So we are very committed to trying to move this forward and get at the molecular level. So I'm going to jump right into our finding. I'll try and give a little bit of background on it, but by and large, I just want to share it with you. And you'll see, I'm really happy that you, Dr. Yamada, was before us. I wasn't aware of your paper until our results came out, but I have a model that applies very nicely to what you just presented. And I really want to stress here, this work is completely independent from the Japanese group. I was not even aware of it, but hopefully you'll see just how complementary what I'm about to show is. So our large-scale RNA sequencing of those cases and controls I showed you in the neural progenitor cells gave us a very, very robust signal. I will give some detail on this, but I just want to say that we do a lot of this across a lot of different diseases, same kind of thing you heard from the Netherlands group, where we have a lot of disorders, including Kabuki syndrome as well, and different ones in the neurodevelopmental space. So we look at a lot of this, we do a lot of genomics, and the signal we saw here was quite remarkable for this particular pathway. And this is the NF-kappa B pathway. I will give you some information on that, but it was really, really robust. It was something we could poke around on, try and prove false, and it just kept coming up again and again and again. And I'll point out that this gene right here, this is the same gene that Dr. Yamada was just showing IL-1B from her, independent mouse studies. Okay, so what we saw is a large increase in the NF-kappa B signaling. So our immediate thinking was, is EHMT1 functioning in the cell to act as a blockade under normal standard conditions to keep this NF-kappa B signaling molecule in check? And I'm going to give you a little, I'll give you a little background on that. So just to show you just how robust these data are, here's across the patients. So here's the matched brothers from our group 1, and here's our missense patient from group 2. This is 2 females, and you can just get a sense. These are just target genes of the NF-kappa B pathway, and this gives you an idea of the variation we're seeing, and it's remarkably tight. These are 2 different families, these are 2 different sexes, and yet you keep seeing this robust effect. Here is Dr. Yama's gene she was mentioning in the last part of her presentation. These are not expressed in NPCs, just to tell you, they're expressed in microglia, And that's what you're picking up here in the control. And yet in this condition, it is quite a strong effect. Something that is supposed to be off is being turned on. So we're really getting a sense that EHMT1 might be functioning to keep this thing in check. And again, this NF-kappa B pathway turns on 400 to 500 different genes, which, by the way, were first discovered in the immune system. This is in NPCs, and when we look in neurons, the effect holds, so that there's a nice replication or reproduction to this. So it's the same cells, but they're differentiated, and you can see the same pathway. There's variation, but the trend holds very consistently across this. And this is also taken straight from the RNA sequencing data. So let me just tell you what this pathway is, so you can get a sense of why I'm very interested in it. So just briefly, I appreciate people are not molecular biologists out there, but it'll give you a sense of why I'm chasing this. So the NF-kappa B are a group of proteins, they're family of related proteins that want to go and do things to the genome. They want to turn on very specific genes in the genome, and they bind a very particular sequence of bases. They need to be kept in a prison cell, essentially. They need to be stopped from doing what they're doing. And if they're not stopped, all kinds of very bad things happen. For example, there's many diseases related to this, which have interesting phenotypic overlap, by the way, with several different features of Kleefstra, including heart, osteoporosis, when it's hyperactive. So the interesting part about this protein is it contains this family of proteins. It has 6 to 7 anchor and repeats. Now that you might be familiar with in the EHMT1 protein also has anchor and repeats. Anchor and repeats are a very common repeat in proteins. 3,000 to 4,000 different proteins have them, but it's the number of repeats that's critical. So the first thing that we found was interesting was that this is a potential binding partner. So what does NF-kappa B do? It is, again, first discovered in immune function. It's known to be important in brain and can change spine density of excitatory neurons. So just to take us back to what we were hearing about from all the different speakers, what I'm trying to propose here, of course, EHMT1 could be doing many things in the cell, but this could be a very important target of what EHMT1 is regulating. And I'm going to try and convince you today that the data are very robust and it's very, very worthwhile to look into. And just to give you a sense of what I have a friend here, what the anchor and repeats look like. Here's all these NF-kappa B family members. This is in purple. And again, these were shown in the cartoons from the previous speakers. Here's what it looks like in EHMT1. And here's these 7 repeats, these anchor and repeats. Here's our mutation in this evolutionary conserve from our family 2. So you get a sense that, and this is a well-known protein binding effect, you get a sense that binding could occur between EHMT1 and NF-kappa B. So of course, the first thing we asked when we saw this large signal is, well, I mean, this seems like something people have shown or would have guessed before. And lo and behold, when you actually know what you're looking for in EHMT1, there's 2 very lovely papers showing interaction of EHMT1 with the main core member of NF-kappa B, the 2 core members, and 2 independent papers. So we already have evidence that this is happening, and this never crossed my radar before because I never knew to look into this particular area. So the first thing we did was, well, do we actually see an association between EHMT1 and one of the core family members? And we did this synthetically at first, just adding tags and doing it in a little cell line, and we can see it very clearly. And when we do it endogenously in in the little boy from group family 1, we can pick up the association in unmodified proteins, which just basically supports what the people that I just showed you on the previous slide were showing, that there is in fact an interaction, and it's probably something that we need to be very aware of. Okay, it's one thing for me to say I found this in 2 people, even if it's matched to their brother, but it'd be nice to show this in different ways. So we spent a lot of time, making cells and engineering cells. So we knocked out EHMT1, heterozygous knockout. It ended up looking like this, an insertion of 29 base pairs, which led to a loss of function. And then we asked, well, do we see the same thing, the same effect on hyperactivity of NF-kappa B? The answer is yes. So you can see here's that same pattern that we were seeing before. We have this significant increase in just the genes we picked as targets. We also did a genetic repair from the little girl from family 2. This is the 948W mutation. And I'll just, for the scientists in the audience, I'll just highlight that when we do our repairs, we always add a silent mutation just downstream. This is in the wobble position, just to ensure that we can in fact show that it is us making this change and it's not just sequencing of wild type DNA. So this is what our traces look like. And it's probably about 80 to 90% pure, so clonal. And here's what it looks like. So same thing in the neural progenitor cells versus the neurons, we see the decrease. So now we're trying to show we can rescue the effect on NF-kappa B, the specific pathway. So we're mutating the mutant EHMT1 back to healthy EHMT1 DNA, and it's lowering the effect. We're comparing it to the mutant itself. This is the only one that does not lower, but this was also our most extreme effect size if you look back at our other data. So it may still be, it's probably still having an effect there. But in any case, you can see just across the board in neurons, we're obviously interested in the neurons because this is the post-mitotic state. NPCs would be more similar to an embryonic brain. So we're seeing the effect probably even more pronounced in the neuronal state. What about pharmacological inhibition? So we want to, if I want to convince you of something, I want to show you through, your own families. I want to show you through an engineered knockout that's isogenic that I've made in my lab using just healthy control cells. And I want to try and show you that if I block it pharmacologically using a known cancer drug, which I have listed here, we can actually mimic the effect of the disease. So just taking healthy cells, we add the drug, we can actually see this increase in all of these same things. It is very, very robust effect. It's unambiguous. We've picked these markers, but you could probably pick a lot of different NF-kappa B response and you're going to see it. This highlights the fact that the histone methyltransferase is probably quite important to this. It's not just the fact that it binds, but the transfer of the methyl group obviously is playing a role because this drug just blocks that. It would not block binding. I can also show you if we look at the same drug, what is the effect on some output markers. First, let me just show you that our drug is working. When we add the drug, it can block H3K9ME2, and you can see as we increase the level of drug, we get decreased H3K9ME2 exactly as you expect. So this is just to show you we're not messing things up. And then come over here and look. Here again is Dr. Amada's protein, IL-1B, which is undetectable in NPCs. It starts to be turned on, and this is protein, and we see a gradual increase in it. Here was that big marker we also saw. It's off when you have no drug, and when you start adding drug, you can, by eye, you can pick out the increase, and this is just a control. So we think these effects are very robust. We see it in the families and the patients, we see it in the engineered knockout, and we see it in just adding a drug to a dish, which is a really nice way to do things because it's an identical cell type. You don't have to deal with all the variation of the cells. Okay, this is an important, this comes up a lot, and we were surprised by this, but I was heartened to see the paper from Hans van Bakkeveen in nucleic acids research in 2016. So I told you at the beginning, we mapped a lot of histone methylation, and this is something we do a lot, and we have a lot of expertise with doing this very carefully. We did this in H3K9ME1, which is not the one people use, although I'm not exactly sure why that is. ME1 and ME2 are both targets of EHMT1. And what we saw just at the ME1 level, so this is what EHMT1 is supposed to do to the histones to do all of its effects. We looked at all the target genes. This is the level of H3K9ME1 we're seeing. And if anything, we're seeing what Hans saw, that there's a slight increase in H3K9ME1. So this doesn't seem to be any genomic effect of EHMT1 loss. This is across the patients and the knockouts on these loci, and I can tell you just across the genome, we don't see it either. Now, H3K9ME2 is in progress, just because obviously we need to check that given what everyone else is saying, but we really, we don't see any difference here, which suggests that the effects are not happening at the genomic level unless it's happening through H3K9ME2, which it certainly could be. I will just finish up with some final data that we have to show you where I think the mechanism's occurring. We pulled out one of these core NF-kappa B family members called REL-A. This is just proof that our IP works and the antibodies working. And very interestingly, I told you at the beginning that the NF-kappa B family, this thing that goes and turns on all these genes, has to be kept in a molecular cage. It needs to be locked down because it always wants to be active. And the role of EHMT1 is to probably help make this prison cell around NF-kappa B. What we found when we did a mass spec, a quantitative mass spec with with both patients. This is something I rarely do. My students always talk about it, but we tried it because of the sort of nature of the experiment. We were actually able to pick up quantitatively. We have a decrease in the prison cell, if you will. The proteins that create the prison around NF-kappa B are a lot lower in the EHMT1 mutation patients. And you can see the other proteins in this family are not affected. It just seems to be the prison cell related proteins that we pick up on the mass spec. And that's again done from an IP of a separate protein. And don't take my word for it. We can even show it just on a straight Western blot. So here's our patients again. You can see very clearly, this is the prison cell protein. It is definitely depleted in the EHMT1 mutation cases. This one is our engineered. This is patient 2, the missense, and this is patient 1, the full gene deletion. It's very consistent. And then again, just to show you the control, here's IL-1B again. Just look by eye. We're just picking up this hint of IL-1B and even CXCL5 and then the loading control. So we think there's a very, very important, robust problem with keeping NF-kappa B repressed. And NF-kappa B could explain a number of different symptoms that children with, anybody has with the EHMT1 loss. So just a conclusion, we think this is a new pathway to explain some of the features of the syndrome. Of course, we're in a cell right now and I'm extrapolating, but if you go and start looking into this, you can start to see the kind of things I'm talking about. We think that the dosage of EHMT1 is critical to keep NF-kappa B in an off state, and that's the family of proteins in human brain cells. It's contributing somehow to building this prison around this transcription factor. We think it might actually interact with several of the members that have the 6 or 7 anchor and repeat domain. We just showed it in 1, but it could be more. We know that the methyltransferase domain is important from the drug studies, but we doesn't seem to be at the H3K9 methylation 1 and still could be at ME2. So we cannot rule out genomic effects yet. And then finally, I would leave you with this, and this is what we're actively trying to work on. We're trying to secure funding now for this. That overactive NF-kappa B is a well-known, it's a targetable gene pathway that many drug companies have drugs for and could be a viable therapeutic strategy in EHMT1. And with that, I will leave you and thank you for everybody for joining, and hopefully we can meet in person in the future years. Thanks a lot.
Thank you, Dr. Ernst. I do have one question for you that just came through, and I'll just repeat it verbatim here. Have you seen any evidence of sustained systemic inflammation in KS patients that could be attributed to NF-kB de-repression?
I don't know, because I don't work with that many. I'm not a clinician, so maybe some of our other folks can answer that. But our prediction would be on hyperactive NF-kappa B or autoimmune related function. We're only in brain cells. I can tell you, when we look in IPS, this effect does not, there is no 0, 0, 0, flatter than flat on anything NF-kappa B related. And maybe it's not expressed there, but it's remarkable how there's just nothing there. So I'm not sure, but what I like about this is it's testable and we can find out easily enough to see if this is something. There's blood tests for this. There's drugs that try and affect it. There's different parts of the pathway that are affected by different drugs. So I think if anything, maybe there's nothing to this, but we can find out and prove me wrong.
Listen, that's part of the process, right? Is to, every discovery or non-discovery leads us further down the road, I think, right? So there's also another question in the chat that I'll let you just look into the chat and answer probably within that. And then we will move on to one final presentation before we take a break. So the next speaker that we're going to have is IDefine's new Chief Scientific Officer. So we had just announced it this week. I am beyond excited to have Eric Sheff join us in this capacity. Eric has been instrumental from the start. So he was one of the co-founders, has been helping all along, but now has a more focused opportunity where he's going to be contributing to the organization day to day. Eric and I speak all throughout the week, every single week. And it's going to take that sort of intensive effort to be able to really push forward our research efforts. So with that, Eric, I'll turn it over to you, sir.
Awesome. Thanks, Jeff. Can everyone see my slides?
We can.
Great. All right. Yeah, so I'm going to give an update of kind of where we stand as IDefine from a scientific perspective at this point. And I will just briefly touch upon what Jeff mentioned, which is I've decided to come aboard and make this the focus of my career. So I'm really excited to do that. Maybe I'm even more excited than Jeff is about having me. And as he said, I've been working on this project ever since we founded the organization, but it was really becoming clear there's so much happening in this space. It's incredibly exciting. There's so many opportunities. And we could see that there needed to be someone to chase down a lot of these opportunities in the scientific space. And so I'm going to be focusing on that. Just super quick introduction about me and kind of where I'm coming from. So I did a PhD at UC San Diego. I did research for years. I then I've done a lot of work kind of in the business world, where a lot of my customers were, pharma and drug developers. Most recently, I was at Illumina, which is the kind of the world leader in genomics. If your Kleefstra family member got their diagnosis by sequencing, I pretty much guarantee it was done on Illumina equipment. So I've been very close to this space, and now I'm going to dive in kind of 100%. And of course, the most important thing of all, in terms of, I think, my qualifications to start working more deeply on this project, is that one of my kids has Kleefstra syndrome. So this is incredibly personal for me, and something I'm really dedicated and excited about. So what I'll be doing for the organization is, well, whatever the organization needs, in short, but a few specifics. I'll be really diving deep and working on building up our research program. So we're really excited about, and I'll talk a little bit more about that, making some sort of targeted investments to push the field forward. Making sure that we have a seat at the table within the scientific community, and that our disorder is well known. Making sure that we're seen at the rare disease conferences, that it's clear that we're out there, and that we want to work with everybody, and we want to plug into the community. And then wherever possible, adding resources. That may mean writing grants, that may mean helping others write grants, that may mean making sure that we're connected to researchers who want to do collaborative work that involves grants, right? So finding ways to bring more resources into our community whenever possible. And then, much more, whatever's needed. In terms of the scientific update, I wanted to start first by just kind of introducing our SAB, our Scientific Advisory Board. So incredibly excited to have all these folks here. Obviously, Dr. Kleefstra needs no introduction. Neil spoke earlier at the conference today, so you've already heard from him. Paul, you've probably heard about Paul before, but if you haven't, so Paul is also a Kleefstra parent, and there he is with his cute little son. And most wonderfully, Paul is a drug developer by trade, so fantastic to have someone like that help us. Allison is one of the world's experts on model systems, cell models, organoid models, etc. And so really happy to have his help as we go forward. Scott is one of the world's experts on ASO technology, which is a really new, exciting technology that I won't get into deeply, but allows us to modulate gene activity. And obviously, that's something of interest to us in terms of that we have a gene that we would like to modulate and control the activity of, since all of our kids don't get quite enough dosage of it. And he was part of the team that got the Angelman's community to a clinical trial, which is, of course, where we want to get. So he's been through the whole arc, right, from the beginning to getting all the way to a trial. And then Stephen Gray is one of the world's experts in gene therapy. So you can see kind of, we've got somebody kind of checking all the boxes for the team. And I just want to say thank you to all these folks. They've already given us so much help and so much feedback and so much information. And looking forward to all the other work we'll do together. So I think we as an organization, we have a lot of goals, right? And part of that is the community, part of that is the clinical, but a big part of it too is, for us, the scientific goal is that when we want to stay laser focused on this, is that what we want to deliver is an effective targeted treatment for Kleefstra syndrome. And Jeff kind of mentioned that earlier, that thank goodness for olanzapine, right? And for other drugs that are used to help our kids. But those drugs are not targeted. Those are drugs that already have existed in the field and are being used for the symptoms. So we want to work to find a treatment that is actually targeting the disorder itself. And I think there really are two big pillars to our strategy. And we've learned this from talking to lots and lots of people. that what you need to do to make this happen, right? So there's two big things we want to make sure we do. And one of them is really you need to make the path more attractive for people who are drug developers. And you need to do that by what they call de-risking it. So what that means is making sure that all the pieces are in place. So if you are interested in developing a drug for this disorder, there's nothing that's really blocking you, right? And I'll get into that in a little more depth. And then also we want to make sure that we travel that path ourselves. We don't want to sit around and wait and say, great, everything's sitting here, so someone come along, right? We want to do that work. And so make targeted investments as an organization to develop and validate early treatment candidates. And those two pieces together, I think, can get us on the right path. And so I want to talk a little bit about that path and what that path looks like. It's a long path, and what you want at the end, right, is an FDA approved treatment. That's ultimately your goal. But there's a lot that goes into that, and it involves really, the whole community working together. And I'll get into that a little bit. So the first piece of that is building up those research assets, right? So we are in the wonderful position as a community that we already have a lot of excellent research that's been done, and you've heard about some of that today, that there's a lot of information on the disease. And you heard from Joost, who's building up some natural history data. We're going to hear later today about more information on what patients are out there, where they are, et cetera. And then I talked a little bit about, you've heard talked about, I should say, earlier today, a lot of work in model systems. So one area we're very interested in is making sure that all the model systems that are needed are out there. And if they're not out there, then we'll make sure that they're created. Because it takes time to create model systems. It's not like you just snap your finger and you have them, but those are important for screening drug candidates, et cetera. And then making sure we are, as a community, developing biomarkers, because ultimately when you get to a clinical trial, you have to decide how you're going to determine if your treatment worked. And that involves biomarkers, natural history information, et cetera. And then we have to get through kind of preclinical research, which is, selecting a candidate approach, right? So that can be drug repurposing, that could be ASO, et cetera. You have to design and manufacture. You have to test those in your models, and then you have to get through safety and toxicology to make sure that that's safe before you can put that into a person. And then you have to go through a clinical trial, which is where you need to select endpoints. And endpoints just means these are the things that we're going to look at, and we're going to say ahead of time, if X changes in this to this degree, we're going to have said we're successful. And those can be behavioral, those can be biomarkers, right? So that could be EEG readouts, it could be all kinds of things. We have to develop those. You have to be able to find your patients, right? Which is why it's important for us to know where the patients are, how many there are, where they live, et cetera. You have to pick out your sites. And then you'll go through phases, and then eventually you submit this to the FDA, and then hopefully you get approved, and then you have your first treatment. And so all of these pieces have to come together, and they have to come together on the right timing. You don't want to get everything lined up, and then at the last minute you realize, well, we don't really know where the patients are, right? So we can't do a clinical trial. And when we talk to drug developers, they grill us, right? Like, well, do you know where the patients are? How many are there? Do you know what endpoints we should use? Are there well-developed endpoints? And so we need to get all of these pieces in place. And then that makes the disorder more attractive for development. And obviously puts us in a position as a community to also do our own walking of the path, as I was saying, and coming up with our own candidates. And because of these have to be, we have to sequence these in time, we're trying to think about this holistically and then work our way backwards, right? We want to get an approved treatment. And so we're going to step backwards through this and say, well, when do all these things have to be ready, and help coordinate the community towards that happening. The other thing I wanted to point out on this slide is that you'll see the little stars on some of these. So all of those are parts where the broader community, and so just kind of a shout out to the community on this call, can make a huge difference, right? So, being involved in providing natural history data, right, for example, and patient information makes a huge difference because it allows the community to understand how they should think about what endpoints there might be for a clinical trial in the future. And so I wanted to kind of end by, talking about that, the ways that the community can help, because it really will take the whole community pulling together, both the researchers and the patient community, to get us to where I think we all want to be. So make sure you participate in these data collection efforts like All Stripes, Rare X, and Genida, and you'll hear about more about those later. I'm stealing Jeff's thunder here, but he's going to talk a little bit more about we're building a new patient locator that will allow everyone to put their information in, and so we can track exactly where everybody is, their ages, et cetera. And then that puts us in a position to go to drug developers and say, look, we know we've already got all the patients, right? We've been told by one that you've got to get to 500. 500 is the number, like the magic number. And once you have 500, then they see it as viable, as something for them to chase. If the community has already been great, I should say also about answering these calls already. Like we had fantastic, response to the All Stripes call specifically. So please keep it up and keep providing more of that information. If there's a need for people to participate for biomarkers or provide samples for models, please answer that call. Help make us more visible. We need to stand out. We need to be a visible disorder. We need to be seen as an exciting place to work. And we've already got this great community, but if we can get even more people interested and expand the community even further, it can't ever hurt, right? So forward things on LinkedIn, retweet our tweets, et cetera. And then, work to expand that network, both make sure, you make sure that you help other people who have Kleefstra syndrome family members get involved. Everyone knows somebody, right? So if you feel like that you know somebody who could help our community, then please reach out, and we'll be happy to engage. So we're really excited about what's to come. We expect to make some investments in the coming year that I think will really be exciting and really help push our field forward. And so stay tuned, and really great to talk to all of you.
Awesome. Thanks, Eric. Again, I think he speaks so well about the kind of the technical points of Kleefstra syndrome, and so to have him on board and having his focus is going to just catapult us forward this year. I mean, it's going to ramp up our efforts 3x at least. So I'm super excited to have you, and thank you for the overview. So what we're going to do now is we're a little bit behind schedule. So we're just going to take a quick 5-minute break. So it's 12:15, 12:16 now. So take 5 minutes and we'll come back online for the next set of presentations around data collection. So we'll see you all in 5 minutes. All right, welcome back, everyone. So the next selection of discussions will be around data collection. So we'll have several coming up. And the first one is Megan O'Boyle from RareX giving us the early results from the Kleefstra data collection program. So Megan.
Hi, everybody. I'm Megan O'Boyle. I'm the patient engagement lead. Joff, can you hear me okay?
I can.
Okay, great. For RareX, which just means that I am the person that works with organizations like yours to get your data collection up and running. My real job is Shannon's mom. Shannon is 21 and has Phelan-McDermid syndrome. We also call it PMS. I didn't name it. Her phenotype is very similar to ones that you're familiar with. She has a genetic cause of autism, ADHD, epilepsy. She is non-speaking. She doesn't feel pain appropriately. She does not sleep unless things are bad. And she has had some catatonia and regression in her later years. I will honestly say that in her 21 years, the first 10, I didn't give a hoot about research. And I'm still not very good about it, but I am blown away by what I've seen today in the few hours that I've been watching. I don't know how long Kleefstra's been identified, but I think it's not 100 years or several decades. And I will tell you, there are rare diseases that have been around for several decades that have not made the progress that you all have and don't have that kind of interest, which is amazing. So you all should be very proud of yourselves and your foundation and your researchers for getting you where you are today. And I have to admit, I'm a little bit jealous. We were identified in 1989 as a syndrome, and we didn't start to collect data until 2010. And it took us 10 years to figure out that we needed to do it better. So my job is to help organizations like yours collect the best data and get it in the hands of all the people that need it as fast and as high quality as possible. So I will start to share my slides. Slides can be boring, and some of what I'm going to show you, you already know. The preliminary data that we've collected in the Kleefstra Syndrome Data Collection Program just reinforces what the phenotype is. So in the questions, that's when it's going to get interesting. So if I go through a symptom slide and you want to know more, I can bring it back up. But I only have a few minutes, and I really want to leave more time for questions from both the families, the researchers, and the clinicians. Jeff, and you can interrupt me if there's a point that you want to make. So later in this deck, I'm going to show you slides like this. Essentially, answers were yes, no, and unsure. And then this one random slide I've chosen has the participant had issues with eyes or vision or ears and hearing. My suspicion is the ears and hearing are ear infections. My own daughter with a similar but different syndrome had ear tubes 3 times. So that's very common in neurodevelopment disorders. The vision issues can be thrombosis or other things. You all are busy, you're tired, and if you're like me, you celebrate if you can get a shower 2 days in a row. So I need you to understand why this is worth your time soon, immediately, but also at least once a year. And one is, all research is great. Some research is limited by the parameters of the institution or the researcher that has been funded. So we're trying to do something that a lot of research can't do immediately, which is return data to you. First, we'll return it to your organization and they'll disseminate it to you. And this is summary data, just like the slide that I just showed you. But wouldn't it be great if you've never met another Kleefstra family to know that your family member is in the 80th percentile? Or wouldn't it be interesting to know that symptom that is causing such disruption in your life is not as common in the other families, that this might be an outlier? So we will return data to you so that you are not left hanging after you answer questions. This is a great way to help identify who would be appropriate for a study. So you have a lot going on based on what I've heard this morning. And it would be fantastic for those researchers to be able to go into RareX's researcher portal and find target patients that would be ideal for their studies, whether it's by location, whether it's by genetics, or whether it's by symptom. And then you will be notified that you may be eligible, and then you can decide whether you want to reach out to the researcher. I'll show you in the next slide the traditional way of getting research data to researchers, and we are going to flip that on its side. RareX will make this data access to researchers around the world in a much easier way than traditionally done. This will also allow you to connect with other organizations or any new organizations that ever come up in the Kleefstra disease, especially if you're out of country. This also lets you update your data, excuse me, your information at any time. My daughter's in a natural history study at NIH. That's great. We go in once a year. We answer lots and lots of assessments that aren't very good for non-speaking people, and then we say goodbye. If she has a new symptom 2 months later or 2 days later, I have to wait until the next appointment. And at some point, the funding will run out on that study, and that will be the end of it. So being able to go in and add changes, whether it's progression or regression, is a huge part of a natural history study. History is the important word in that, is you want to see progression over time of a disease. And that is why we will remind you to come back and update things. We do not own your data. IDefine does not own your data. Dr. Kleefstra doesn't own your data. You own and manage the data. You decide how widely or broadly you want your data shared. And hopefully all of this will lead to drug research faster. I just wanted to give you a heads up that on your beautiful website, if you go for new families, newly diagnosed, under caregiver community, you'll see join RareX. And when you hit that, you go to this beautiful community page. You see Kleefstra up in the left-hand corner, one of your beautiful families members. And right here is the Get Started button. And you'll see at the very bottom, I've highlighted the translate button. And it's one of those translations where you click on a flag. This community page is your front door. It will explain to you what the data collection is, why you should participate, and has frequently asked questions. And like I said, you can translate this. But once you hit get started, at the moment, this is in English only. That will not be forever. We will be adding 2 more languages at the beginning of 2023. This has to be global, and it will be global. You actually have a larger disease than many of the diseases that we're working with. We're building this for all 10,000 rare diseases, but we've decided to start our focus in neurodevelopment. So of the 25 groups we've already brought on, you are one of the 23 that is a neurodevelopmental disorder. What do you need to get started? You need a computer, email, and a password. You don't need all your medical records. You don't need a detailed family history. You don't need a lot of time because you don't have to finish this all at once. You can work on it until somebody wakes up, or till the bus comes, and then you can come back. We do say use Google Chrome or Apple Safari. We're definitely showing issues with the other browsers. And although this is designed to work on all mobile technology, phones and tablets and such, we're not crazy about the user experience right now. So I would recommend that you not use them, but use a laptop or a computer. So this is just an example of one of the questions in the very first survey after you have set up your account and your consent. And that is I call it head to toe. Do you have brain issues? Do you have behavior issues? Do you have growth issues? Yes, no, unsure. These are mandatory questions. There's about 24 of them. It covers every system of the body. That was all we had up until this last month. We've added 19 new surveys. So those of you who have already enrolled, you've got lots of surveys waiting for you, and we want you to come back. You will only see a survey, though, if you say yes to a question. So in this dashboard, you'll see in the gray is your completed surveys, and in the blue is your in progress. And this person is working on diagnosis, and down here under level 2, behavior, vision, and growth. These are showing up here because they said yes to something in that first head to toe survey. So here's some results. I'm going to fly through these. there does not seem to be a high incidence of cancer. And in terms of head, neck, and face, in the question itself, this includes any dysmorphic features. So that may be where the 34% of yes is coming from. So eyes and vision, this is a high incidence. And like I said, in your community, I think it could be thrombosis. Ears and hearing, not surprising if you have a lot of ear infections. Participant has issues with growth, 32%. And I don't know enough about your community. Phelan-McDermid syndrome was considered to have tall patients until we had enough of them. And then we realized it was just happened that the first few families happened to be tall. And that speaks to the fact that you need as many people in a data collection as you can get, because the numbers will show what the true phenotype is. Has the patient had psychiatric issues? Not surprisingly, this is a high number. Cartilage and bone would probably involve orthotics and other needs for correction. No surprise about digestive system, and in lungs and breathing, there may have been some incidents of pneumonia and things like that. For all of these, and I can come back to some of them, the point is, if you said yes to something, you're going to get another survey, a level 2. And that level 2 survey is not going to be, it's going to take you 5 to 15 minutes, and it's still going to be pretty high level. So if you say yes to brain, then you'll have a brain neuro survey. And in that, it may ask you about sleep or seizures. It won't ask you what kind of seizures. And if you say yes to seizures, then in a few months, when the seizure survey comes out, you will then have a more detailed survey. By owning and managing this data, you are able to say how broadly or narrowly you want to share your data. So far in your community, 61% have said, I will share this data de-identified. We don't share any identified data. I want to be very clear with that. I will share my de-identified data with any type of researcher doing any type of research. That could be socioeconomic research. It could be research on ethnicity. The other 39% want to share their data with health, medical, and biomedical research. Either is fine, and you can go back and change your answer anytime. So far, 79% of Kleefstra syndrome respondents have said that they are willing to share their name and email with any organizations. Again, if IDefine or other organizations have your email, then they can keep you up to date on things like conferences and researchers and things like that. So I'm not going to show you all the demographics, but not surprisingly, because we're still in English, there's 39 in the United States. We've got some in United Kingdom, go Brazil, that's awesome, and Canada and so forth. In all, in our first 10 disease communities, we had answers from 46 countries. This is not surprising at all. I just wanted to show this. This is typical of a lot of rare genetic syndromes, and this has nothing to do with the genetics and everything to do with the socioeconomics of genetic testing and who is given genetic testing. So those of you who have already enrolled, so you followed Jeff's and Julie's call in the previous year and you enrolled, that's fantastic. I just showed you a bunch of charts about what you answered. You need to come back because we have this great new tech platform with lots of improvements and lots of new surveys. So you need to, Jeff will provide this to you all somewhere. Log back in, you'll have some new terms to agree to. You'll have to reset your password and reconsent. Once you do that, you'll have access to all those new surveys. This is what I was saying earlier. The traditional form is a researcher has to know your disease exists. And remember, you're one of 10,000. And as our previous speaker says, you all need to stand out. Then they have to know you have some data, and then they have to request it with all sorts of applications and jump through hoops. Your foundation then has to decide whether they're worthy of getting the data, and then they have to send it. And then the research is done, and who knows what comes of it. Sometimes you find out and sometimes you don't. This all takes a lot of time, money, and effort. RareX is taking lots of diseases and they're all entering through one door, like the one I showed you with the beautiful girl jumping, that community page. But then their data goes to this place. And then the researchers on the right go in and set up an account and provide credentials, and then they can query. And they can query on genetics, on symptoms, or disease. If someone to go into RareX now and query on intellectual disabilities, autism, and epilepsy, they would be getting data from probably 19 of the 25 groups that we currently have in there, or actually probably even more. So this federated data access is going to get more eyes on your data. It also will allow people who have never heard of Kleefstra syndrome, like you were a few years ago, to stumble across your data and maybe realize that this is data that they're interested in. I'm going to stop there just because I want more questions than slides. Jeff, are you manning the chat or do you need me to?
Yes, I just had a question come in that I answered, but you can probably expound on it. It was referencing whether there is a need to come back to the surveys kind of year over year, and is that going to be a functionality? And perhaps you want to kind of, allude to that regarding that.
If you don't do it, it's a snapshot. And a snapshot is nice. You all want history. You want progression of disease. When did things get better? When did things not change? Not changing is really important. Even if nothing has gotten better or worse, you still need to come back. And we at RareX will remind you, but I'm pretty sure Jeff and the rest of the team will remind you also. But you don't have to wait till the 1 year mark. If your child unfortunately starts having seizures or experiences some regression, you log in, you change your answer that was formerly no to a yes, and now you have a new survey on your dashboard. If we come out with a new survey, for instance, a level 3 on seizures, and you don't see our email that says, hey, we've got a level 3 on seizure, please come back. When you come back at the year mark, you'll see that that new survey is on your dashboard. I can't be more clear about this. This is your opportunity to show data across the life of the disease. And it doesn't have to be once a year, and it doesn't have to be for the 5 years of funding that research usually gets. This is meant to go on, and it's only going to be as good and as deep as you all make it. And to our previous speaker's point, the researchers don't want to come shopping if the shelves are empty. So we don't want to publicize that this data is available if people aren't finishing the surveys or they're not enrolled. So I know you don't have a lot of time, and I understand that you are tired of surveys, but this is really critical. This is not a replacement of all of that other cool stuff. This is not instead of Genida. This is not instead of all of those other surveys. This is complementary to it. And hopefully someday in future, when somebody wants to study Kleefstra, you can send them to RareX and say, okay, these people have already answered 60% of your questions, so just ask them your new ones. And that way we're not burdening you. But we are a nonprofit. We're not selling the data to industry or anybody else. Nobody's paying for this data. You all aren't, your organization is not paying to have it collected. We are doing this to get to treatments, and we had to remove the burden on groups like you, and we had to remove the barrier from getting high quality data that regulators and industry need. And these are standardized questionnaires. They won't say, does your Kleefstra child have? Because it's not about just your community. It's about all the similar communities, because that's where the research is going. There's things called bucket trials and basket trials and all sorts of other things where drug trials can go across disease. But this is not just you doing a favor for researchers and industry. I can tell you the first 10 years of my kid's life, I didn't care about research. I didn't think it was going to happen in our lifetime. And it was very overwhelming and confusing. Still overwhelming and confusing, but it's happening now. It's happening faster than we ever thought. And that data that we collected, and I got that aggregate summary, I took it to IEP meetings. I showed my mother-in-law, who didn't think my kid had anything wrong with her. And I got doctors to order tests that they were not likely to order because my child didn't show symptoms. But when I could show the school team or the medical team that there was data, not crazy mother, data, then they listened. And with a lot of research, all of this great research that's happening, it takes years, and then it has to be published. And there's that, it just takes time. It's hard. And so we can't always afford to wait for those publications to get our one-to-one on our IEP. So this is going to give you something. It's also going to educate you about the things that you haven't thought about. We're asking about every system in the body, not just what we think your phenotype is. And I can tell you personally, after 10 years of data, when my daughter started to have catatonic issues because of Phelan-McDermid syndrome, we had zero data because nobody had ever asked about it. So nobody ever studied it and nobody knew about it. So I strongly encourage you, as you can tell, I get very passionate about this. If you ever feel like there's nothing that you can do for your child, this is something you can do. This will change things. Data sounds boring, but it moves things along. And as two speakers ago said, they're working on 2 patients, and that's showing huge progress. But we need data on hundreds or thousands, depending on the size of your disease. Jeff, I'm going to stop there, and you can ask more questions, or I can.
No, I think it's, I love how you speak passionately and just transparently for everybody watching. Megan has been a soundboard for me and a huge assistance as we've launched IDefine. I mean, prior to IDefine, I was touching base with Megan because of the examples and the connections she's made within the rare space. And so she has been guiding us not only from, the industry perspective, but also as a parent, and you can hear that. And so she knows what we're walking, she knows the path that we're taking, and she's here to help us. So just a big thank you. There is one clarifying question about whether the general research and medical research, like what is the difference there? So maybe Megan, that you could answer that real quick.
Sure.
How do we make the data most accessible? Is it for general research, or is it the medical and bio research?
For the type of research that you all want done, either is fine. I am happy to have anybody, including somebody doing, hypothesis building, who's a math major at Stanford and running numbers in the middle of the night. I'm happy for them to see some sort of pattern that nobody's looking for. But there are cultures and ethnic groups that have been burned, and they don't want people doing any research other than medical, and that's fine. The other question that is asked on the next page that you can skip is, I want all research except commercial, which means I want everybody but pharma. And that's your choice. I personally want drug companies to have my child's data. And the final question is, I want the researcher to have had an IRB approval, which means whatever institution they're working for, they've had this ethics review that says this is good data. This is a good study. That's perfectly fine. I chose not to click that box. Again, I would like to get the data out as broadly as possible. Remember, de-identified data. We're not showing anybody identified data. Your own organization will not see identified data, but they will get this aggregate summary data that they'll get out to you. And that should help you be able to be a better medical advocate, even if you're not interested in research. So it's not just for research. I said, it's for you too. Researchers are excited about this because it's helping them, and industry is excited about this because they want high quality data, but they don't want to have to slow down and go collect it. They'd rather have it done this way. So I encourage you, we'll be collecting genetic reports. You'll be uploading genetic reports in the near future. That's as important as coming back every year. There are so many diseases that look alike that researchers have gotten to the point that if they don't have genetic proof, they can't assume that the person isn't just self-identifying without a real diagnosis. So you will get lots of messaging in a few months about that. But I just want to say you all should be so proud of what your organization and your researchers are accomplishing. It is amazing, and you are way ahead, but that doesn't mean you can slow down. So if you can go raise funds to get more done, that's fantastic. If you can answer surveys, that's, even better or equally better. So if you have any questions, please let Jeff know. But for all of those people that were early adopters, thank you. But only 20% of you have come back. I need all of you to come back and update your account so you can see all those new surveys.
Awesome. All right. Thank you, Megan. And I think it's, there was another question added here that I'll just answer really quick about how many confirmed cases of KS are there. And the direct answer is we don't know. And the only way that we're going to find out is for everybody to raise their hand and participate in these surveys. So there's guesstimates out there right now that actual diagnosed are around 1,000. But there's prevalence. There's the hypothesis that the prevalence of KS might indicate that there could be 250,000 patients out there. But again, the only way for us to really identify is for folks to contribute to these data collection efforts. And just being a competitive person that I am, when we were part of the early adopters and provided that opportunity, I want us to be the #1 condition. If we're the #1 condition in all stripes, in Genida, in Rare X, then that is going to create noise and create visibility for our condition, and people will want to work with us more. We can't underestimate how powerful that is. So to piggyback off of that, Megan made a point about the collaborative effort in nature of Rare X and all stripes and Genida. One is not independent of the other. We are looking to build this kind of collective, deep resource of information and complementary. And so it's now time to bring in the team from All Stripes, so Ashley Leek and Angela Jackson, and present some of their findings as well.
Great. Thank you so much, Jeff. And thank you to the community for letting us speak today. We're really excited to share what we've been working on with our Kleefstra Syndrome community. And it's last year, it was so great to see everyone come together and join. And so now we're excited to give things back. So I'm going to share my screen really quickly. So we're going to be talking about how you can accelerate drug development in Kleefstra syndrome, and we've been having the blessed opportunity of working with IDefine to really hone in on our Kleefstra syndrome research and grow our cohort. So a quick introduction as to who we are is my name is Angela Jackson. I'm currently the Senior Kleefstra Program Manager at All Stripes. I've been working at All Stripes for about 3 years now, so I have the absolute pleasure of working with Jeff, Julie, Eric, and all the wonderful team in IDefine and just the Kleefstra syndrome community in general for really understanding how we are engaging with the community, how we can continue to be better advocates, and how do we make our research relevant. And then I also have Ashley Leek with me. Ashley, if you want to say a quick introduction.
Sure. Thanks so much, Angela. Hi, everyone. Thanks so much for your attention today. I'm Ashley Leek, a member of the research team here at All Stripes. My background's actually in brain cell biology, but in my last year at All Stripes, I've been really interested in understanding the unique challenges facing the rare disease community, like the burden of traveling for care, the time to receive an accurate diagnosis, and the burden of identifying excellent care providers. So some of the data I'll share later on gets at some of these challenges.
Ashley. So today we're going over our agenda. First, I'll go over our mission and our story to really understand what is All Stripes and then how it works. And then Ashley will be going into more of our Kleefstra syndrome research that we've been doing for the past year. And then we'll go over an FAQ to answer any frequently asked questions that we have ready. And then of course, feel free to continue to ask questions that we'd be happy to answer either aloud or on the chat as well. So to really understand what All Stripes is, we really love to go over who we are, what we do, and why, and that's embedded in our mission, which is to unlock new treatments for people affected by rare disease. And this mission and the people behind it are why I'm here, and I'm proud to be here today because I see this mission embedded in every person that we work with, every patient advocacy group that we work with, and then also our wonderful ambassadors and patients as well. We know that we can't do this alone, and so All Stripes works to bring together communities, advocacy organizations, and researchers and drug companies to develop drug development and research faster. So we're empowering all stakeholders to work together to create a better future for those impacted by rare. We also like to go over our story. So All Stripes was founded by Ano and Nancy Yu, and Ano is actually a rare disease patient himself. He has neurofibromatosis too. And what he found as diagnosed as a patient, he realized that his medical records contained a powerful story. And so what he wanted to do was take this story and provide it to researchers so that there could be advanced treatment for his condition. The problem was that as he was signing up and joining all these researches, he found that his data often got siloed and that it was just staying within the places that he signed up for. And what he was frustrated was on is how can we bring all the research together and push something forward. And so this is so relevant to what Jeff was saying about creating a collaborative effort for research. So this is what he founded All Stripes on, is we aim to give patients complete access to their medical records, really learn from these powerful data sets, and then allow patients to participate in research and track their research from the comfort of their own home. And so how does All Stripes actually work? So for those of you who are new and aren't on our platform, All Stripes is a research platform dedicated to rare disease. And so how it works is it's a user-friendly platform where patients or caregivers can sign up on behalf of their patient. It takes around 15 minutes to sign up. And then during the sign-up process, patients and users sign an electronic consent form, and this allows for their accounts to be created. And this umbrella research consent allows us to use their de-identified data for minimal risk research and surveys and recontacting for additional research. And of course, Ashley will go into more of what research is actually being done. And then also within the sign-up flow, patients can list their facilities, and then All Stripes will do the work from there. All Stripes will collect these medical records and organize them and put them into your platform for you to easily retrieve and access as well. And I know a lot of patients talk about how when they're compiling their medical records, they have this thing called the binder, which is just a huge binder filled with medical records from all your different facilities. And I know that patients are seeing more than one facility at a time. And this really makes it easier for them to have a complete access to their records in real time. And so records are requested once a year to make sure that all the information are kept up to date. Because of course, once you sign up, you're still going to get care, you're still going to see facilities. So we want to make sure that the records are most relevant and up to date and represented in our research. And of course, All Stripes allows you for recontact for future study and recruitment when needed. And so currently now we have records and data collected from over 3,800 facilities within the US, UK, and Canada, and that number is just continuing to grow. Currently, we are only available for getting medical records for the US, Canada, and the UK, where we have where our electronic consent form allows for, but we're still working on seeing where we can expand and be helpful across other countries as well. And so as you know, the medical records are a rich source of information about a patient's diagnosis, their symptoms, results, and how the condition is continually managed. So the information that we collect from these medical records, and then through things like patient-reported outcomes, we have surveys where we ask patients and caregivers their experience or their symptoms. We're able to create these real-time insights, which again, Ashley will go into more depth on, but combining these research insights will allow us to work with researchers, clinicians, as well as the patient community and foundations to make sure that the things that we're collecting is relevant and can help answer new research questions for the community as well. And then we provide these aggregated de-identified insights, both to share within social media or within the foundation, and then also within the platform itself as well, where we have this thing called your care journey, where you can see your most common symptom and then also compare that de-identified to the rest of your community as well. That's on the All Stripes platform. And then, of course, if you signed up and you're thinking, what else can I do? We want to make sure that patient voices and the patient experience is still centered in the content that we create. And so we have our ambassador program, which really works to bring people together to raise awareness and advocacy for their condition. And so we encourage families or patients who are willing to talk about their story and their experience to consider being an ambassador as well, where we can create a lot of content around stories or social media posts or anything that can continue to bring awareness for your condition, because the more people that are hearing about your condition, the more that we hope that this will inspire researchers in the community to continue to push forward as well. And of course, with patients in a rare community, some communities, they feel isolated. And so hearing people go through similar experiences as they can really bring the community together as well. And so I will pass it off to Ashley to give a quick update on the research that we're currently doing with our Kleefstra community.
Great. Thanks, Angela. Can you go to the next slide? Great. So today I'm going to present some of the data from our Kleefstra syndrome cohort at Allstripes. In this first slide, I'm just showing like a high level overview of the demographics of this cohort. We currently have 78 participants in research, where 47% are male and 53% are female. And the majority of this cohort resides in the United States, but about 15% live in either the UK or Canada. This cohort is representative of ages spanning 0 to greater than 30 years of age, which is shown on the graph to the right. And 23 patients are between 3 and 5 years old. And the majority of patients are younger than 12 years old. Can you go to the next slide? So on the previous slide, we saw that the majority of the Kleefstra syndrome cohort is located within the United States. This slide just gives like a little more information about where patients are actually located in the US. So the majority of Kleefstra patients in our cohort live in the South census region, which is highlighted here in pink. And about 86% live in large metropolitan urban areas of greater than 50,000 residents, which is shown in the graph on the right in pink. And this data is fairly consistent with the geographic distribution of the general US population, although we do have a higher proportion of patients living in the South and Midwest and slightly more located in urban areas. Next slide, please. So the rest of the data I'm going to share in this webinar comes from this symptoms survey that is in the care journey section of our platform. Because it requires participants to fill out the survey and only includes patients with a full set of records available, I'm only going to be able to share data from 25 patients in the cohort. Here at Allstripes, we always do our best to protect patient privacy. So in some of the following slides, I've grouped patients to best protect them from re-identification. Next slide, please. All right, so in the survey, we asked patients or caregivers 3 different questions. The first was the year at which symptoms began. And the x-axis of this graph shows years of age in single years, such that ages 0 to 11 months are shown in that first bar on the left and 1-year-olds to the right. All of the answers to this question were in these 2 age groups. The y-axis shows the number of patients who began showing symptoms in each of these age ranges. And we found that the most patients in this cohort began showing symptoms before their first birthday, which is highlighted here in blue. Next slide, please. Okay, so we next asked patients what year they were diagnosed, or caregivers, what year the patient was diagnosed. This graph on the left shows the age of patients when they first received a diagnosis of Kleefstra syndrome. The x-axis at the bottom shows the different age groups, starting at 0 to 1, and increasing age every 2 years all the way to greater than 14 years old. The y-axis shows how many patients fall into each of these age categories, and the bar highlighted in pink demonstrates that most participants in our survey reported that the patient was diagnosed with Kleefstra syndrome between the ages of 0 and 1, which is great. Although there's a lot of variability in that age of diagnosis. In this graph on the right, we looked at the year of diagnosis, and most patients were diagnosed in the last 7 years. And particularly the most patients were diagnosed between 2017 and 2019, as shown by those 3 middle bars. Okay, next slide. So using both the reported age at diagnosis and the reported age at symptom onset, we could measure how long it took for a patient to receive a Kleefstra syndrome diagnosis after their symptoms started. So experiences varied really widely in the cohort, which is indicated by the hash marks at either end of the line. Patients in this cohort received a diagnosis 0 to 27 years after symptom onset. Next slide. If we look at the box on this graph, we can see that the middle 50% of patients in the cohort were diagnosed between 1 and 5 years after symptom onset. Next slide. The line in the middle of the box represents the median, and this is the halfway point for all of the patients. So the median time from symptom onset to diagnosis was 3 years, meaning that half the patients were diagnosed less than 3 years after symptom onset, and the other half were diagnosed more than 3 years after symptom onset. Next slide. Okay, so to understand the initial presentation of Kleefstra syndrome, we next asked survey participants what was the first Kleefstra symptom that they noticed. We found that 36% first noticed developmental delays, which is shown in the graph in dark blue. And 28% noticed hypotonia, which is shown in medium blue, which is low muscle tone. And then 36% experienced other first symptoms like feeding difficulties, dysmorphic facial features, hearing, or visual impairment. The most common first symptoms, developmental delays and hypotonia, are really consistent with the medical literature suggesting these are characteristic features of this syndrome. Next slide. Okay, so finally, to understand the burden of this condition, we asked survey participants which symptom most affects quality of life. 84% responded that developmental delays or intellectual disabilities have the greatest impact on quality of life, which is shown here in the graph in the 2 different yellow colors. This was followed by a developmental regression, behavioral difficulties, and other symptoms. So we hope that in the near future, we will be able to provide deeper information using data we found in the medical records. And we've already actually collected all that data. I personally just need to get to making the report so that we can share it with you all. So hopefully in the next couple of months, we'll be able to share that back. We're really grateful to all those patients and families who have already generously provided information for this study. And we look forward to providing more insights soon. So thanks so much, and I'll pass it back to Angela.
Thank you so much, Ashley. And then now we're going to go over a couple of frequently asked questions as well, just to align really on some questions that we often get asked. And then, of course, feel free to ask additional questions that we may not answer currently. So how does Allstripes keep patients' data safe and confidential? So the only people are very secure and are not accessible records or share their account with a caregiver or with a trusted researcher. I think I'm going unstable. Okay. And their doctors, then that's okay with them as well. But regularly the patient account is secure. And also the if a patient chooses to be a part of research, then of course their informed consent allows for Allstripes to share strictly de-identified information. So any other data will not be shared unless it has been properly de-identified. And then who owns the information and data in the medical records that Allstripes collects for patients and families? Similar to RareX, we believe that patients always own their own data, always. So patients who do consent to research can also opt out at any point. And of course, if they just want to get their medical records all in one place and then opt out later and just download all the medical records that was collected, that's completely fine. Of course, the patient always owns the medical records that we collect. And so it is their information, it is their data. If patients and caregivers consent to being a part of Allstripes researchers, our researchers will organize the de-identified data in the from their medical records to combine with others within the community that also allowed for research as seen through Ashley's wonderful insights, and then analyze them to look at the data forward to help advance treatment. And then there's a question of do I have to pay to participate in All Stripes? And no, we believe on making sure that it's our commitment to patients and families that it is never a cost to join All Stripes, and it is not a cost to get your medical records into your account. The other questions ask if you will we let you know if we share your de-identified data? And of course, we will notify you and let you know that when your de-identified data is going to be used a part of research, and make sure that you are consented to be a part of research as well. And then we'll also notify patients or families if there are any additional research opportunities if they want to be a part of that as well. And then finally, who can access the research? So if the patient and the user gives proper research consent by signing our broad electronic consent form, then the research community, people like academic researchers, clinicians, or within the industry can work with All Stripes to access strictly the de-identified data for research purposes. Of course, as in many of the presentations, we believe that the more people that are researching your condition of interest, the better. And so we will never enter any exclusive partnerships with a specific researcher or with a specific company, because given ANO's commitment to making sure that data isn't siloed, we also want to make sure that we're working with everyone who is interested in working with our data. So of course, there might be other questions, but if there's anything that we don't touch upon today, please feel free to email support@allstripes.com for any questions, and we'd be happy to respond to you then as well. Thank you.
Thank you, Ashley and Angela. Really appreciate it. And there was a comment in the chat regarding whether to do either or on several of the surveys. And just to quickly answer that again, like, we want this to be as collaborative as possible. We're not positioning one organization ahead of the other organization. They're complementary. And I think the biggest kind of point of differentiating is one is medical record collection and the other is patient reported outcomes. So those are very differing, right? Not everything in the clinical medical records, in the notes, or even in just a basic survey is going to be the same of what a parent can report from day-to-day experience. And so it is incredibly important to be able to have both of those. We have heard from day one that the deeper the data set, the better for us. And that's going to come into play not only for the researchers, but once you get into FDA and trying to make sure that they know that we've done our due diligence there. So one other question just came in. Let's see if we can get an answer to that. Are we always made aware of who is accessing the data? That's a really good question.
Yeah, so of course, if any of the de-identified data is used, we want to make sure that that is properly run by the patient, because of course, at any point, a patient or caregiver can revoke their consent. And so to be able to have that opportunity to opt out, they also have to know when they are going to be opted in as well. So if there are any new research opportunities, we want to make sure that the community is aware and that they know who we're working with as well. So if at any point they do want to opt out, that's completely optional as well. There also is the opportunity to opt back in later. So if, say, there is a research opportunity that you don't want to be part of, you can revoke your consent and then possibly reach out to us again as well if you want. There is a research opportunity you do want to be a part of. Good question.
Great. Thank you all again for being here and for answering the questions, and looking forward to just kind of the continuing engagement with you as we work together on making some progress here. So thank you all.
Thank you.
Thank you so much.
So again, Dr. Kleefstra actually mentioned this in the chat here, that there is, the repository of information within Genida. So Jean-Louis and Pauline, both there. So again, we've connected all these players in the data collection space. So the Genida team talks with the All Stripes team and talks with the RareX team. So we're really kind of triangulating the data to make sure that at the end of the day, we leverage the need for accountability and responsibility and collaboration between folks that we work with. And if folks are going to be siloing any information, then it really does not behoove us as we move forward. So that is kind of one of our core values, and I'll allude more to it later, about being relentlessly collaborative in all of our efforts. And I think you see that with our data collection partners. I think you see it with the research partners within our community, as well as cross disease. You know, we're looking at neurodevelopmental disorders as a whole. We're looking at other conditions that express similar phenotype, and particularly and potentially kind of underlying genetic machinery similarities, where maybe, you know, inroads in one condition could lead to insights and progress for another. So that's the way we have to work and operate, and we're doing that. So next we're going to have Tanya Zdolsek-Droxler present. So Tanya is an amazing mom of a KS daughter, Anya, from Slovenia. And so Tanya has worked with IDefine for a while now, but she's also works in the data field and machine learning and AI in her career. And she's also recently just launched the Kleefstra Syndrome Europe Alliance to try to bridge some of the communication gaps that exist across countries within Europe. So she's going to be presenting some of her findings from reviewing the GENIDA data.
Hi. My name is Tanya Zdolsek-Droxler, and I'm from Slovenia, Europe. Some of you already know me, some don't. In short, professionally, I'm working as a research project manager at the Jožef Stefan Institute here in Slovenia. But I'm also a mother to a daughter who has Kleefstra syndrome. And that is why I'm also active in advocating Kleefstra syndrome and have been collaborating with IDefine and Kleefstra syndrome organizations from Europe. I'm mainly interested in data management, data science, and research in general. Today, I'm going to present first data analytics results for Kleefstra syndrome based on the GENIDA data. So the online questionnaire of GENIDA has 46 major questions with sub-questions and presents medical information, behavior, development, quality of life, and so on. And it's also important to state here that the data is anonymized. To start, some demographic data. So there are 172 cases reported for Kleefstra syndrome in the GENIDA. Regarding gender, there's almost a balance, as we have 90 female cases and 82 male cases. Regarding the age, we have participants ranging from infants to 45 years old patients. And looking at the country of residence of the participants, we see that we have 48 cases, for example, from USA, then 18 from UK, France 17, Germany 15, and a lot of European countries, but also Australia, Canada, Brazil, Colombia, Japan, Egypt, also India, South Africa, New Zealand. So based on this, we see that GENIDA is really a global registry. Here I have to add that we have also 19 cases where the country was not specified, so we don't know the country of origin. So as already said, GENIDA has 46 questions, and the first 5 questions are containing open answers. So these are sometimes hard to analyze with basic statistics. So basic text analysis was used to get some meaningful insights. So we did look for combinations of 2 and 3 words to see which words often show up together. So for example, question number 2, what's the major behavioral cognitive problem that affects your relatives and the family life? We have 165 answers, and the results show gets frustrated, intellectual disability, nonverbal, behavioral issues, lack communication, impulse control, mood swings, unable to express, makes difficult, new things, play alone, short attention span, grabbing, pinching others, express feelings, pain, adult constantly, moderate intellectual disability, constantly to entertain. Going on, question number 3, did your relative suffer a major medical problem or discomfort following specific medication? Here we have 164 answers, and the results are showing side effects, negative behaviors, rinklonopin, statin drugs, extreme agitation, catatonia, no, increased negative behaviors, issues, trazodone, agitation. So I have to add here that this is an overview of first results, but as I don't have a background in medicine, we still need to consult with a clinician, a medical doctor, regarding specific results. Moving on, question number 4. What are the major medical problems that occurred until now? We have 152 answers, and they are saying ear infections, hearing loss, sleep apnea, epilepsy, febrile, seizures, sparing, severe psychosis, hypotonia birth, low muscle tone birth, recurrent middle ear infections, moderate hearing loss, inconclusive hearing test, open heart surgery, problems air liquid, fibrillation severe birth, aspiration significant. And I'm also happy to report about question number 5, which is a little bit more positive. So please state your relative strengths. And we have 150 answers, and the results are this. Strong-willed, good memory, very curious, happy, following, laugh, play, great sense humor, happy, eager things, loves music, singing, trying new things, will to live, challenging task, encouragement, loves, learn, try, seems good, numbers, singer, dancer, wonderful, science, follows, routine, others, loves, eating, positive, extroverted, happy. Moving on, we have here the answers around weight of our children, and at the beginning we have the number of answers, and for example, first we can see in that there is a little bit decrease of answers. So for example, for the weight at birth we have 142 answers, but then for the weight at 6 months we have only 79 answers, and then also for 1 year 72, for 2 years only 64 answers, and so on. And for 10 years we then have only 26 answers. So here I have to add that Geneida is not a one-time questionnaire, but you can come back and again provide answers for specific questions that you think needs an update. So if we here look at the mean value, we see that at birth, our children do have 3 kilos and 202 grams in average. The next question is problems during pregnancy. This is a very important question, and we have 174 answers. And the parents did have to choose around 3 answers. So problems during pregnancy, I don't know yes or no. And the majority of answers is indicating yes, we have problems during pregnancy. We had problems during pregnancy. So we got 109 answers stating yes. And further on, the ones stating yes were further on asked what words best describe them. And they had to choose between different answers. And the majority did choose excessive amniotic fluid, reduced fetal movement, cesarean section, and other. And the ones who choose other were then also asked to specify these problems, and we got these answers and results: irregular heartbeat, emergency cesarean, umbilical cord, premature rupture, rupture membranes, heartbeat prompt, emergency, stuck birth canal, wrapped around neck, reduced heart rate. So these were 3 answers, and we prepared the text analysis again. Going on, we have developmental milestones given in months. There are 4 of them: sitting without support, standing independently, walking, and first words. And for sitting without support, we have 130 answers, and the mean value is 13 months. Going on, standing independently, we have 100 answers, and the mean value is 24 months, which is 2 years. Regarding walking independently, we have 109 answers, and the mean value is 30 months. And regarding first words, we again see a decrease in answers, so only 90 answers, and the mean value is 27 months. So from my personal experience, I also had some difficulties remembering about the first words of my daughter. So sometimes it's easier to watch some old videos to remember, or check old health records where these milestones are sometimes also stated. Communication ability. The question was, does your relative communicate in any of the following ways? Multiple answers were possible, and the most chosen one were vocal sounds, noises, gestures, with speech. Regarding understanding, the question was, does your child understand what's being said? Here we have 160 answers, and the majority, so 121 answers are indicating yes, our children do understand what's being said. Further on, the question is your relative's speech likely to be understood by? And there are 4 possibilities: family, family and others who meet your child for the first time, neither the family nor others, and others who meet your child for the first time. And the majority of 153 answers did state yes, my kid's speech is likely to be understood by the family. So the family and others who meet your child for the first time, for this around 30 answers were positive. Regarding diagnosis of autism, so here we have 166 answers, and the majority, 97 answers, are indicating that our children in the majority don't have autism. Then behavioral problems. So here we have 156 answers, and the majority of answers indicating if our children do have behavioral problems. I don't know, no or yes are the answers. And the majority, 104 answers, are indicating yes, our children do have behavioral problems. And for those who stated yes, had also to choose what words best describe this behavioral problem, and they had this full of behavioral problems. So aggressiveness, self-aggressiveness, obsessions, phobias, diagnosed schizophrenia, impulsivity, hyperactive, attention deficit, shy, anxious, depressive tendencies, restricted interests, repetitive behaviors. And for each of them, they had to, so the parents had to state the seriousness. So if this problem is mild, moderate, or major. And for example, we generated this graph, and for example, for mild, we can see that around 40 answers did indicate to have mild self-aggressiveness, around 32 did state mild aggressiveness, and around 25 mild repetitive behaviors. So this was really a short overview of the first results. We will continue to work on the analytics, and the results will also be included in the Kleefstra Syndrome Clinical Guideline that is being prepared under the supervision of Professor Kleefstra. So to conclude, I can say that the future is bright for rare diseases, but data is still crucial. So we parents can really help to improve the clinical care and the research around therapies. As in each of these fields, data is needed, and we can help with sharing information about our children. And this kind of data is called patient-reported data, and it's crucial in rare diseases. So I did state here all of the options for contributing this patient-reported data for the Kleefstra community, and we are using 3 of them. So besides Janaida, we also have RareX, and then we have also All Stripes, which is handling electronic health records in USA, Canada, and UK. So thank you for listening. If you have any questions, new ideas about data, you can also email me. Thank you.
All right, thank you, Tanya, for that presentation. So now we've kind of been able to see this holistic snapshot of where data is, the importance of it, and again, it's a lot of information, but I think it's going to yield insights into our patient community, and hopefully, as we continue to contribute to it, we'll be able to yield some of the answers that we ask as our child ages, and we can start getting a little bit more informed insights about how how Kleefstra syndrome impacts our kids. So next we're going to be jumping into some clinical perspectives. So we have 2 more presentations in that regard. One is going to be coming from Zoe and Dr. Sid from Boston Children's Hospital, and then we'll also be having a report from Emma Palmer down in Sydney, Australia. So I will turn it over to Dr. Sid and Zoe.
All right. Awesome. Let me just pull up the slides. Okay, well, hello, everyone. It is such a pleasure to be a part of this conference and to have the opportunity to speak to the Kleefstra community. My name is Zoe Frazier. I'm a genetic counselor at Boston Children's Hospital. And today, Dr. Siddharth Srivastava, also known as Dr. Sid, and I will present updates on the Kleefstra Clinic at Boston Children's. So overall, we are a multidisciplinary clinic that provides evaluations and clinical care to children with Kleefstra syndrome. We aim to serve as a resource to the Kleefstra community, but it's also important to highlight that we learn so much from each family that comes to our clinic. Each family contributes to our wealth of knowledge, and we feel really privileged to have the opportunity to work with such an engaging community. So the main providers in our clinic are Dr. Sid and myself in the neurology department, and then Ann O'Donnell is also a geneticist who works in the genetics department. Our clinic is a part of a large hospital, Boston Children's, and we work to harness the resources and collaborate with other providers at Boston Children's. And so we have partnerships with other specialties such as psychiatry, sleep medicine, gastroenterology, and ophthalmology.
Great, thank you so much, Zoe. For those who don't know me, my name is Sid Srivastava, and I'm one of the neurologists at Boston Children's, focused on neurogenetics and neurodevelopmental disabilities, and I help run our Kleefstra clinic here. And it is, once again, it is such a tremendous honor to be able to talk to you and to learn from all of you, from everybody here. And I'm very delighted to hear all these wonderful talks today. I wanted to go through characteristics of the patients we've seen so far in our clinic. I hope this information may be helpful, may be useful to give a general sense of some of the challenges and needs of the community at large. We looked at data over the past year and a half. Most, though not all, of the patients are captured here in the slides that I'm going to show you. I'll show you data on 44 participants. Over 2/3 of these patients are girls, while about half, or 1/3 are male. I've also plotted age ranges of the participants, of the patients. The greatest age range comes from infancy to early childhood, followed by age 6 to 10, age 11 to 5. This number declines with older age groups. It's not surprising that I think more younger kids tend to come versus older individuals. Interestingly, there were several individuals we saw ages 21 to 25 and even some individuals 26 years and older. And next slide. I also wanted to give a sense of where the families are coming from to see us. The gray represents states where we have not seen patients, but shades of blue represent number of patients from different states. Light blue is one patient from that state, slightly darker is 2 patients, and so on, all the way up to very dark blue being 7 patients. We saw the most patients from the state of Massachusetts, which is not terribly surprising given that's where we are located. We did see a number of patients from some Midwest states like Illinois. Next slide. Now I wanted to talk as well about some of the clinical characteristics of the patients we have seen, starting with genetics. 37% of the patients we've seen have a chromosomal deletion that includes EHMT1. The remaining have a non-chromosomal or intragenic defect affecting EHMT1. I've listed the specific types of intragenic misspellings. So 32% have a frameshift misspelling, and that's basically where there's an insertion or deletion of several letters that lead to a bunch of erroneous amino acids that are built into the protein, followed by a premature stop signal. 2% have a missense variant, that is a substitution of one amino acid for another. 11% have a nonsense variant, which is when one amino acid is substituted by a premature stop signal. 7% have had a partial gene deletion, that is a portion of the EHMT1 gene is deleted. And 11% have a splice site variant, which affects how the coding and non-coding portions of the gene are arranged. Next slide. So what about the cognitive characteristics of the families we've seen? The vast majority have intellectual disability, which is defined as having an IQ more than 2 standard deviations below the mean, or global developmental delay, which is a label we sometimes use in younger kids under the age of 5, sometimes as a precursor to the intellectual disability label. Notably, there were a few families we saw whose children had IQs in the non-intellectual disability range, which is quite interesting, I think. In other words, ID is certainly commonplace in Kleefstra syndrome, but it's not universal, it's not 100%. Next slide. What about behavioral challenges? By far, the most common behavioral diagnosis we saw was autism or autistic features, affecting 40% of the patients we've seen. This was followed by attention deficit hyperactivity disorder. Other challenging behaviors include aggression, binge eating, and OCD, obsessive compulsive disorder, just to name a couple of examples. Next slide, yeah. So we looked at mood characteristics as well. Among mood characteristics, anxiety was relatively common, affecting close to 20%. Depression also was relatively high. It occurred in about 10%. There were a few individuals with catatonia as well as psychosis and hallucinations. And although I didn't mention this, actually I'll call this out specifically, in terms of regression, I think about 3 out of the 44 families we saw reported a history of regression, which is a low number. It's actually surprising to see that, but there could be, it just could reflect the fact that we're not seeing kids over the life, or we haven't, at least not yet. We're seeing families for the first time initially, and maybe there was regression that happened earlier on, or and that wasn't, well captured. So just a caveat about that number. Next slide. Actually, Zoe, could you go back to the previous slide? Yeah. So the topic of seizures comes up a lot, has come up a lot in our discussion with families. Interestingly, not that many patients that we've seen have presented with the diagnosis of seizures or epilepsy. 7% had a diagnosis of epilepsy. And remember, epilepsy is defined as having 2 or more unprovoked seizures in one's lifetime, or having one unprovoked seizure with the high likelihood of having a future unprovoked seizure. There was an equal percentage with a history of abnormal EEG without clinical seizures. there was also a small number of individuals who had febrile seizures. Next slide. Okay, great. I've listed some of the other neurological challenges that families have reported and experienced. Sleep challenges by far are quite common. So this includes obstructive sleep apnea, periodic limb movements of sleep, and just reports of generally disrupted sleep. So altogether, these sleep challenges are seen in maybe about a quarter of patients, which may be an underestimate, I think. CVI or cortical visual impairment affected 40% of the patients we saw. Dysphagia, which is trouble with swallowing, coordinating those movements, affected 16%. And microcephaly, which is defined as having a small head size, was seen in 18% of the patients we saw. Next slide. So finally, here is a list of the various types of systemic challenges that have been seen by our families that we've seen. The vast majority have had GI challenges. They include constipation, reflux, and so forth. Many have had cardiac issues, including congenital heart defects. I won't go through each of these categories, but needless to say, there is a wide assortment of systemic challenges in addition to the neurodevelopmental and neurological and behavioral features that we see. Next slide. So this is, of course, a very high-level overview of the common set of challenges that patients who come to us have reported. Once again, I really want to emphasize how much it's a privilege, how delighted we are to learn from each and every one of the families. And we're so gracious and so thankful for everybody's time, effort, and being able to learn from each and every one of you. And it really inspires us to do our best to try to optimize clinical care. So once again, thank you to each and every one of you, to the families out there. Great, thanks.
Awesome. Thanks, Zoe and Dr. Sid. Just fantastic. It's amazing to think of first meeting y'all in 2019 and how we've been able to work together to build this incredible center, and having that kind of responsiveness from the community, being able to visit you guys and you all being able to provide the support you do is amazing. And so certainly appreciate both of y'all and all of your efforts. And so next we're going to be shifting to the other side of the world with Dr. Emma Palmer. And Emma and I have connected a number of times. There's several families in Australia, most notably for me, the Corey family, who I've engaged with quite frequently and have done a lot of work to boost our efforts down in Sydney. And so Emma is just a powerhouse and amazing lady. And they are working or have launched a Kleefstra Clinic down at Sydney Children's Hospital. And she's going to be providing a overview of their work down there.
So it's a great pleasure to be able to join this amazing international virtual conference today to talk a little bit about what's been happening recently at Sydney Children's Hospital in Australia to try and support families who have a child with Kleefstra syndrome. My job as a clinical geneticist is one I feel incredibly humbled to do so. I'm a doctor who trained as a pediatrician, and I see my role as to listen to families who have a child with a suspected rare genetic condition and see how I can try and address their questions, their concerns, and be part of a journey with them to make sure they get what they want, which is the best possible outcomes for their child. And I think that starts with listening to families. There's a lovely quote from this book: "When in doubt, ask the experts, those with a lived experience and those whose voices have not been heard." And I think sadly, so often families who have a child with a rare condition don't have their voices heard. They're just struggling to get through each day, trying to get across the difficulties of living with a child whose symptoms don't make sense, who have a child with a condition that hardly any other doctor or allied health professional has ever heard of. And if I can be some small part of trying to join those dots together, but also connect up families with amazing global communities like this one, so that families can learn that they're not on this walk alone, and that I can learn and share from them and hopefully pass on that wisdom to help other families. So we have a beautiful little girl here. Very grateful to the Corey family for sharing pictures of the beautiful Gabby, who has Kleefstra syndrome and has absolutely amazing parents who say, this isn't right. This isn't right that we should have such a struggle to get a diagnosis and to get the support and care and the opportunities for research and clinical trials that our child deserves. And we hear you, and we would love to continue on this journey to try and get better solutions for all families with a beautiful child like Abby. So a little bit about Australia. You may tell from my accent that Australia is not my birthplace. I was born in the north of England, but I've been very lucky to live here for the past 20 years. And as of this week, we have 25,856,834 people in Australia. That's one birth every 1 minute and 45 seconds. But I also want to make the point that we're a very multicultural community, and one person arrives to live in Australia every minute and 1 second. We have a history that goes back many, many, many millennium before the migration of people from Europe. And there is an incredibly complex and wise society of Indigenous and Torres Strait Islander people here. And like I mentioned, we are a very diverse community now, and a quarter of our population is born overseas. Mostly Australians live around the coast of Australia, and we have a state-based health system with different states having slightly different approaches to health. What all of these things put together means is that healthcare is not yet equal across Australia. For those living in rural and remote areas, for those of Indigenous heritage, and for those of culturally and linguistically diverse groups, there are more barriers to accessing excellent healthcare, and that's something that we want to work very hard to tackle, to make sure that every child with a rare disease gets the best possible care and support. In 2020, Australia launched its National Strategic Action Plan, which was endorsed by the Australian government. And it talks about the vision of the best possible health and well-being outcomes for Australians living with a rare disease, which is 3 million Australians. Critical enablers are listening to families living with rare disease and working in partnerships with our governments, our state and national governments, but also looking abroad for global collaborations, and to try and understand how to provide the most high quality, comprehensive and integrated healthcare for those living with rare diseases that truly is person-centered, which means tailored to each individual person. I work at Sydney Children's Hospital, which is part of Sydney Children's Hospital's network. Together, these are two children's hospital sites across Sydney, and we make up the largest hospital network in Australia. This is where our hospital is, very close to the ocean and also very close to the city. We're developing as a hospital network with expertise in digital health, and the pandemic has shown us that we can pivot to effective telehealth, which often means we can connect up with families, avoid them having to come into the hospital, but also connect up better with their local healthcare providers so that they are really empowered to work in partnership with families. And we're also very much at the fore of advanced therapeutics in Australia, which means things like gene therapy. And we were the first site in Australia to give gene therapy for spinal muscular atrophy and for childhood retinal disease. That's a blinding eye condition. And across the two hospital sites, we see about 2,000 new families a year with a suspected rare condition like Kleefstra syndrome. And we've listened to families, and what we hear is really the ideal is to have a multidisciplinary clinic for rare disease. So one where we can act as a centre of expertise and a point of contact for the local medical team, so the local primary physicians, paediatricians, gastroenterologists, neurologists, so that they can share and understand the really world-class expertise in a rare condition like Kleefstra, but also to be a point of contact and support for families, and also connect those families for those individuals where they want those connections in informal and more formal ways. And also really critically for those families who are interested to know about advances in research, get our families what we call clinically trial ready. So to be in the best possible position to not only attract clinical trials, but hold successful clinical trials as new therapies come on board. So we've been working over the last few years about a framework and a structure to put all of our families, all of our rare disease families in the best possible position to get excellent care when they have a child with a rare disease. It starts with our patient registry called GeneStart, which helps us identify cohorts of families. So that means groups of children with a particular rare disease like Kleefstra syndrome. It also helps us when children don't yet have a very clear-cut diagnosis to tap into research opportunities locally and internationally to fast-track them to a diagnosis. And that's part of our program called GeneAd. And then where we'd really like to go is to putting all these things together to have these multidisciplinary clinics that offer the best possible care. And over the last couple of years, we've actually been quite successful in getting some funding for parts of this, in particular our patient registry and our undiagnosed disease programs. So with Gene Together, we wanted to start somewhere, and we started with Kleefstra. So our current Kleefstra clinic is very much held out of our genetics department. I'm lucky enough to work with Caroline Chalou, who's a senior genetic counsellor in our unit, Dr. Lisa Ewins, a clinical geneticist, and Rebecca McIntosh, a genetic counsellor. And we all are affiliated with the university, so we have an academic role as well as a clinical role. Lisa is also at the Garvin Institute, and I'm also leading a national program which is about trying to improve the understanding and awareness of doctors and allied health professionals about rare diseases. And all of these we bring together to try and put us in the best possible position to support families. And this is the idea, is that families can be referred to our Kleefstra clinic actually from anywhere in Australia. So when they are referred to our service, we will ask if we can collect information not only about their genetic diagnosis, but all of the investigations that have happened before, because it can be exhausting going through and telling the story again. So we want to try and shortcut that as much as possible by gathering the information that has already been done by the local doctors and the local teams. And then through seeing the families in our clinic, we can let them know about patient support groups. We can also collect all of that information together in a letter and identify if there's any gaps, if the families are not meeting any of the recommendations in the management guidelines for Kleefstra syndrome as they currently exist. And we're also part of the international collaboration to further refine those guidelines. So if there are some gaps identified, we can help either refer those families to the services that they need or let their local health teams know what the referrals are that we would recommend. And really critically, we're understanding that I think every single family we've seen is interested to learn more about research opportunities. So we start that by offering them enrolment into GeneStart, which gives them us the permission to allow families to connect up with clinical research databases and enroll them in research trials as those come up. But also really importantly, it helps us to understand how we can do better. So as part of Gene Start, we have the option of families joining focus groups or having interviews so we can understand what we're doing okay and where we can do better. And also by being on our registry and being connected in to this telehealth clinic, we can let them know about new research opportunities as those come along. So for example, for another group of patients who've got genetic epilepsies, we've held a couple of family days, which have been really successful in joining up families, and we'd love to do that for Kleefstra soon. And we have various smaller projects that come along the line, and by enrolling into our registry, we can let people know about that. So for example, for those with epilepsy, we're running a current pilot where families can learn and ask the questions they want to know about their child's epilepsy or the management of their child's complex condition. So we started towards the end of last year, and we've been lucky enough to meet with 10 amazing families and 4 more booked into our next clinic. We started with children across our network, but we've extended that out, and actually the hospital is happy for us to see families from all across Australia because there is no other equivalent clinic yet in other states. So our genetics counselling team will usually ring the family before. As I said, that will help fast track collecting all of that information so that we don't need to spend so much time in the appointment going through everything that's happened before, but also really importantly ask the families what is it that they would like to get out of the clinic, what are the questions that they have that they'd like to be addressed. As I mentioned, we then check that the management guidelines are happening and are there any gaps where we can help advise their local teams regarding specialist referrals. And we inform them about our patient registry and research opportunities. We then put all that information together in a letter that goes back to the family and their local clinical team. So it's really about trying to empower that local team. And we're also happy to help with patients navigating things like the disability sector, referring them on to appropriate support organisations and offering them a review as well. So here are some themes. My first theme was that our families rock. It is humbling to meet every single family and hear about what amazing advocates they are for the people in their lives with Kleefstra syndrome. Sadly, we're hearing about very varied access to specialists. Some have got very engaged, often a general paediatrician who's really good at referring them to the supports they need, and others that's been more of a struggle, which is where hopefully we can step in and help that process. And time again, we hear about how hard it is having a loved one with a rare condition and having to retell that story every time and feel isolated. So hopefully our clinic will really importantly try and bring together some community of families to reduce that isolation. We also know it can be really hard in Australia to explain to our disability and education sectors what having a rare condition like Kleefstra is. So this is somewhere where we can particularly help with, for example, letters to the National Disability Insurance Scheme to try and make sure that children are getting the appropriate therapies that they should be and should be appropriately funded for. We're also growing our little black book, as I like to call it. So when we hear about a hero specialist, a wonderful gastroenterologist, a fabulous sleep physician, a really good psychiatrist, then we note that down, and that's someone that we might recommend to the next family we see if that's a gap in the care for their loved one. And like I mentioned, almost universally families are interested in connecting up with other families and in hearing about research. So our hopes for the future is that we might be able to secure some funding to have a true multidisciplinary clinic rather than seeing the families and then referring them on to specialists, to actually bring all those specialists to the family, which would be the ideal situation. And also we're so excited, we're bringing other advanced therapeutics trials for other conditions, as I mentioned, into Sydney Children's Hospital Network, and we really want to make sure that we're putting our families in the best possible position for those clinical trials. As I mentioned, we're really looking forward to developing a local family day so that families can connect, hopefully when this pandemic is settling down and we can have the opportunity of actually meeting face to face, which would be wonderful. And we would love to grow what we're learning from this process to be a model of care that we can apply to other children, other families with other rare neurological and neurodevelopmental conditions. So thank you very much for your time listening today. That's my contact details, elizabeth.palmer@unsw.edu.au. And please don't hesitate to reach out if you've got questions. And I'd love to connect and hear from more of you. Many thanks.
Thank you to Emma for sharing her story from Sydney, Australia, and just a couple other notes. There is someone else in Australia that worth mentioning, Angela Morgan, who works a lot on on speech. And so I know that her and Dr. Kleefstra and the team at Radboud are working on developing a trial for speech around Kleefstra. And she's done this with a couple other conditions, SET BP1 that I'm familiar with, as well as Coulin de Vries, and really understanding speech within our patient population and how to improve it. And then on another note, I mentioned the Khoury family previously, but there's actually a pharmaceutical company that Emma and Vanessa down there are engaging with that have developed a drug for several other conditions such as Rett syndrome, Pitt-Hopkins, Phelan-McDermott, and are in a clinical trial with them. And we feel like there might be an opportunity to repurpose a drug to also help our community. So a lot of really fascinating work happening in Australia. So now we're going to basically bring this to a close today. So I appreciate all of you who have hung around till the end. I want to share just a few quick updates before we wrap. But again, I think I began with gratitude today, and I want to make sure that I really emphasize and close with gratitude because the time and the effort spent on creating this conference was not minimal, and it's very appreciative from Zoe and Dr. Sid to the whole team at Radboud, all the presenters, Julie, Drake, Tanya for helping. So I'm going to just quickly share my screen and just reinforce what our mission here is at IDefine. And we're committed to identifying life-changing treatments and cures for those with Kleefstra syndrome. And so our goal is to do that through three separate realms, and it's community engagement, it's coordinated care, and it's research. So, you know, how we do that is going to be, my apologies, is going to be several different kind of efforts, and it's going to take really an entire team. And one of the exciting things that Eric alluded to earlier, and I want to give a huge tip of the cap and expression of gratitude to Haluk Akin, a father out of Turkey who has worked with his business partner, Seda, on building the Kleefstra Syndrome worldwide map. And so this is now live on our website today, where we're going to be able to really get a firm count on how many patients we have. We want this to not be an IDefine effort. This is not a kleefstrasyndrome.org effort. This is not any individual organization that's working on KS. This is a global effort to understand where our patients are, how many do we have, and we could utilize this in several different ways. One of which could just be very simply, hey, there's someone with Kleefstra syndrome that lives within 30 minutes to me. Wouldn't that be wonderful to know, to get together and do these small regional meetups? I think it would be pretty amazing. And so if everybody could go to the website and utilize this new function, it would be really, really incredible for us as a tool moving forward. In our mission, we always want to keep the why directly in front of us, and the why is our loved ones. And so it's typically the part of the conversation that I get choked up on, because I am a father to a young girl with Kleefstra, and she's my world. She means everything to me, and I know that your loved ones do the same for you as well. And we have to keep their voice and their needs front and center, because oftentimes we are the advocate, we are the voice for them. And the way that we're going to keep them front and center is through the data collection. That is a representation of their voice. We also want to engage our community through conferences like this, as well as the conference that we're hosting in person outside Chicago in June. So if you haven't registered for that, another tip of the cap to Becky Libby and Mindy Kramer and Mike Goff and Julie and so many other people who are helping build this incredible conference. It's geared towards our patient community and their loved ones about how we can, facilitate and support each other. Another kind of tip of the cap to Laura Slamkowitz, who helps up heads up our community engagement, and who just recently hosted a webinar on teens and adults with Kleefstra. And I hung out for the first 5 to 10 minutes and got to see Eden and Julie and others talking back and forth. And it was just super powerful. It was really cool to see that interaction. And so we're hopeful to continue to add more of those events through our It Takes a Village series, through Laura's effort. So if you just email us at community@idefine.org, we can start building out programming together. Let us know what's important to you. And again, helping facilitate some regional meetups as the world starts opening up again and we can gather with one another. So, highlights of what this past year has been. So, we launched in 2020 on KS Day, and it has been an absolute sprint of working as hard as we can to bring treatments and opportunities to our patient community as quick as possible. And so a couple of highlights, if you receive our newsletter, we just showcased our efforts with Combined Brain, which is a collaborative effort with 30 other conditions, where we're pooling our resources to be able to push research forward faster. With All Stripes, we're able to partner with NCATS, which is a subdivision of the NIH here in the US, about understanding what is the cost of rare disease odyssey and how that diagnostic odyssey impacts. What are those hidden costs associated to try to further the entire rare disease community? Candid is something that I'm really excited about, that we're just announcing today that IDefine is going to be co-sponsoring. And what it is if anybody spends any amount of time in our Facebook pages, that GI and cyclical vomiting syndrome and bowel movements is a hot topic. It might be the most talked about the most talked about topic of discussion that we have. And so Candid is an effort to further understand that. And so in fact, Megan, who talked earlier from RareX, Phelan McDermott, is going to be kind of one of the main sponsors, but it's also working with researchers out of Indiana University's health system to understand what's taking place in the world of GI, and especially with kind of cyclical vomiting syndrome. So there's going to be a survey, there's going to be a conference at the beginning of June. So again, I think subscribe to our newsletter, follow us on social media. We'll keep you informed about the events there. But I think one of the takeaways that I wanted to convey is your voice matters. The topics that you're discussing, I'm trying to monitor as frequently as possible, because we want to, we're not in this for our own selves. We're in this to help ease the burden on you and on our loved ones. And so the more that we know, the more we can kind of make informed decisions about where we act and where we invest our resources. And then on the kind of industry recognition, Megan alluded to it, and we've heard this before, that we're a pretty young organization and that we're a pretty young disease as far as discovery is concerned, but we've been able to grow by leaps and bounds. And recently we were recognized by Global Genes as being rare champions of hope. And that is because of our entire team that's been contributing efforts and pushing us forward. And then Eric, who you heard speak earlier, who's joined us as Chief Scientific Officer. Eric and I have just recently been invited to Ultragenics, which is a big player in the biotech space, in the pharmaceutical space, for rare diseases. And Eric and I are incredibly fortunate and excited about attending their rare disease boot camp. And that's going to give us the tools and the network to push our effort forward even faster. So we're going to that at the end of April. And again, it's with the patient voice and our community in mind of how we can push this effort forward faster. Our team, again, this is just a few of the folks that we have contributing their efforts. So Mario and Matt on communications, Julie's driving partnership efforts, Ashley on the grant coordination, Laura on community engagement, Derek and the rest of our board really kind of helping, be the sail that's kind of guiding our journey. And then, again, the conference team, Haluk, international leaders like Sebastian Ziegler, his wife Ines, Vanessa that I've mentioned down in Australia, Michelle, and then also kind of the other partner organization leaders. So Wendy from CleefstraSyndrome.org, it really is an entire international effort. And without the contributions of everybody, we're not going to move as fast as we need to. So thank you to everybody there. Thank you to the TNC, the team at BCH, Radboud. Every time I've picked up the phone or I've emailed somebody, your responsiveness has been incredible. Kira, Maya, all the folks at BCH have just been amazing thus far. Our goal, just to put it out there, is we're seeking to raise $2 million to fund what we're trying to do. Research is not cheap. So we're working through grants. We want to raise $1 million to invest in some of the research efforts with the coordinated care centers, like the one in Sydney, like the one at Radboud, like the one at BCH. That's not enough. We need to be able to have equity and accessibility across our patient community. So how do we develop that? We need to make investments. We talked with Dr. Kleefstra on Kleefstra Syndrome Awareness Day that focus takes funding, right? So unless we're able to fund these things, then other conditions could potentially pass us by, or they will get the focus necessary. So we need to raise funds for that. Community development, the conferences that we host, especially the in-person conferences, are going to have costs associated with them. And as much as we can help offset those costs to the patient community, so as many people can come as possible, so we can bring people in. I know Zoe and Dr. Sid have open invitations to come to Chicago this summer. And then lastly, the innovation piece is the rare disease landscape is rapidly changing. And so the more that we can be flexible and pivot as new opportunities present themselves, it will behoove us in, again, being one of those leaders of the rare disease space. So with that, I'll bring the conference to a close. But again, thank you to everybody. If you need anything, if you want to join the effort from a volunteer capacity, if you want to host a fundraiser, I'm hosting one next month here outside of Greenville, South Carolina. And I know the Reed family has hosted a walkathon out in Kansas, and people have hosted dinners or wine events. There's a myriad of ways to host events to bring in fundraising efforts. And so if you need any support there, that's what we're here for. Any other questions, any other opportunities that you see, any other needs that you have, bring them to our attention, and we will do our best to join you as we walk the journey together. So with that, I hope everyone has enjoyed today's conference, and we will see you soon.